Good afternoon, everyone, and welcome to our first BC COVID-19 Virtual Research Symposium, jointly hosted by the BC Centre of Disease Control and the UBC Faculty of Medicine. I'm Rob McMaster, the Executive Associate Dean of Research at the Faculty of Medicine. I'm really grateful that you've had the time to join us to hear about COVID research that is happening across BC. The goals of the day are to hear about the recently funded COVID-19 research, to enable collaborations to continue and be established, and to share resources across the province. We are at full capacity, having 500 participants now, and the symposium is being recorded and will be available next week. We anticipate that this is the first of many of research meetings on COVID over the next few months. I would like to acknowledge that UBC and the CDC are located on the traditional territories of Musqueam, Squamish, and Tsleil-Waututh peoples. It's worth reflecting on the fact that joining together remotely means that we are gathered on traditional territories throughout the province. For now, for some of the housekeeping issues, I'd ask that you keep your cameras off, and this keeps the bandwidth lower and keeps the speed continuing along, and your microphones muted. I would like to start by introducing our co-host, Dr. David Patrick, and it was his idea to have the COVID Research Symposium, so really glad to be working together. David is the director of the UBC Centre of Disease Control, and at CDC is the director of research and medical epidemiology and the lead for antimicrobial resistance. David is also a professor in the UBC School of Population and Public Health. David?
Good afternoon, and thank you, Rob. It's a pleasure to introduce our opening speaker today. You know her in many guises, but she started off professionally as an officer in the Royal Canadian Navy, trained in public health and preventive medicine, was with public health in Ontario for the SARS epidemic. And not too long after that, we were lucky to recruit her out here to British Columbia. During her work at CDC, she did many things, but had a prescient focus on planning for pandemics, and later crossed the Salish Sea to eventually land her current gig. So it's my pleasure to introduce our opening speaker, Dr. Bonnie Henry, our Provincial Health Officer and Clinical Associate Professor at the School of Population and Public Health at UBC. Bonnie, if you're, you may need to unmute there, Bonnie.
Hey. Okay, there we go. Thank you very much, David, and I won't take very much time, but I just wanted to say how we are in unprecedented times, and having spent 30 years planning for pandemics and having lived through some pandemics with many of you over the last 2 decades, it still is a shock to me what we've done and what has happened over the last few months here. One of the things that we started very early on in this, in January, was making sure that we put the building blocks in place to be able to understand what was happening from a scientific perspective. And I just want to raise my hand to David, and to Mel, to many of the rest of you who are on the line from the BC CDC, from UBC, and researchers from across the province who are connected across Canada and across the world, so that we can better understand how this epidemic, this pandemic is affecting us on many different levels, from the population level to the laboratory diagnostics and transmission levels, to the clinical, the important clinical trials part of it, and of course the important pieces of developing a vaccine at some point. So this is a fantastic initiative. I've been so proud of the way that we have come together as a research community here in BC, and really I believe are leading the way in how to work collaboratively, and how the work that you do informs the decisions that I am part of making on your behalf on a day-to-day basis through this. So thank you very much. I am not going to be able to stay for all of it. I have to do my little thing today, but I will be getting a report of this, and I'll be talking to many of you over the coming weeks and months as this progresses. And I'll leave it at that. I just do want to acknowledge that I am on the traditional and unceded territories of the Pathumban-speaking peoples here in Victoria, particularly the Esquimalt and the Songhees First Nation, and it comes to me a lot that we need to think of doing our work in a good way, and we rely on, very grateful for the keepers of this land in helping us focus our attentions on how we can best use the information that you're gathering and you're pulling together to look after our families and our communities here in BC and around the world. So thank you.
Thank you very much for that, Bonnie, and thank you for working tirelessly to lead BC's response to COVID-19. We're very proud of you. I'm going to spend the next few minutes, people, just contextualizing BC's research response a little bit. In essence, we've been playing defense against COVID-19. For a history buff, it feels a bit like the Battle of Britain, except this time the few to whom we owe so much today are our frontline colleagues in emergency and critical care and at the grocery store. Their ventilators, in a way, are our Spitfires, their protective equipment is their ammunition, and sometimes it doesn't feel like we can have enough of either. We remain early in the pandemic, and we say this because we are nowhere near a threshold of exposure where immunity in our population can work to our advantage. Without public health measures and distancing, many more people will be infected and our health system overwhelmed. Yet during this viral blitzkrieg, there is already hope. It's not too soon to plan for an end to the pandemic and to conduct the research that will make that possible. You've heard that the curve is flattening. Nobody in their right mind will consider this a permanent turnaround unless those numbers persistently decline and we do all the right things to keep it down. But there is a game plan for victory, and it requires research. As Dr. Henry announced last week, we formed a Strategic Research Advisory Committee to work with the province's health emergency response structure. We need to focus on research questions that, when answered, help us plan for an end game to the pandemic. And in support of this, the Michael Smith Foundation for Health Research COVID-19 Research Response Fund will focus on high-quality research that informs the ongoing response. The plan looks kind of like this. First, we need to achieve control over transmission by pulling together to sustain our efforts. Yes, more of the same with respect to distancing and things. Once transmission becomes rare, we may be able to carefully relax some measures while maintaining others, and such decisions can be guided by epidemic modeling, rendered safer by borrowing feedback tricks from our process engineering colleagues, and will involve complex decisions that involve not just epidemiology, but economics and human behavior, so that data science and analytics and other disciplines play vital roles. A fuller victory will require that we take the fight to the virus. We will most likely need a vaccine, a highly effective treatment, or another strategy to reduce transmission to zero. Now, we all know that Canadian vaccine candidates are in animal studies, and novel treatments designed by BC researchers are being evaluated in clinical trials. When this is over and the threat of transmission wanes, we've got to learn from experience and retool. What could have led to a faster global and local response? What investments in public health services, science, and laboratories will assure we're far better prepared the next time? And most important, I think, as alluded to by Bonnie there at the end, I think, what will we have learned about ourselves? Today, we are working as one to pull through this difficult early phase. Shouldn't it be easier now to understand fair wages for long-term care workers and flexible care for substance users? When the end game plays out, and it will, we must not forget what can be achieved when we remember this communitarian spirit. So we're going to hear from some of the researchers who are working towards those solutions. And I just want to benchmark that since first gathering with a small number of these folks in person on February 13th, researchers, including those speaking today, have accrued $9.5 million in funding to get that job done. So before starting up with our first bank of speakers, we've just got a little bit of housekeeping to do. We will run today as a series of 5-minute rapid-fire talks within 5 sessions. We'll start with diagnostics and genomics. We'll get into clinical trials and patient management. We'll then cover epidemiology and public health response. We'll talk about social dynamics, communications, and trust. And we'll talk about the work ongoing here and elsewhere around therapeutics and vaccines. Now, as Dr. McMaster pointed out, this is our very first time running such a large session via Zoom. So thanks for your patience as we adapt to this new way to connect. Some housekeeping notes then. We expect that we will be at capacity today. If you're stepping away, please log off to let others take your place. We may need to remove people to make room for speakers to join us later, and we're sorry if that happens to you. This session is being recorded, however, and will be available in full next week. Questions will be accepted via the chat feature. Please message Michelle Wong if you have a question and indicate who the question is for. If we have time at the end of each session, she'll ask 1 or 2 questions, and we're certainly encouraging our speakers to finish with time to spare for questions. Now you'll note that you're all muted. Please also leave your cameras off to maintain higher video quality for everyone. If you use a UBC VPN, please disconnect from the VPN and rejoin. For the best Zoom experience, we'd like to recommend using the speaker view. And lastly, if you'd like to receive emails to share outputs from this meeting, details about future meetings and information on COVID-19 research, please sign up for the mailing list. The URL is in the chat and will be posted between sessions. So of these speakers, we asked 4 questions for you today. They are, who are you and where are you working? How is your research responding to provincial or global health needs? What do you see as the key gaps in COVID-19 research? And what resources do you have that can be shared among this community? So let's begin to hear their answers. Our first session is on diagnostics and genomics, and it's a pleasure to introduce Natalie Pristajecki and Mel Cragdon from the BC Centre for Disease Control, who will open up with Steve Jones. They've got 2 5-minute sessions because they've got a lot of projects together, this group. So that's 10 minutes for them, and they'll be followed by Peter Unrau from SFU. So I think we need to make sure that Natalie and Mel are on. They're both together on Natalie's screen.
Thank you, David. I'm going to start. It's Mel Cragdon. I just want to talk about some of the provincial assets that we have in terms of nucleic acid testing. We have a capacity to test about 5,000 samples a day, 7 days a week. Those tests are distributed across BC. The sites are listed. There's point-of-care nucleic acid testing, which is going to be delivered to various rural sites, particularly those that have transport challenges or immediate testing needs. We are following the medical health officer directives on who to test, and at the present time we have a reserve capacity of about 3,000 tests per day, 7 days a week. There's no testing backlog, and there's the turnaround time is about less than 24 hours from receipt to result. We've been working very closely with the Genome Sciences Center to have contingency plans for swabs, extraction reagents, amplification reagents, and this has been a very fruitful collaboration. We have some IT solutions that enable us to now integrate all the testing parameters hourly or daily in BC by location, and these can be linked either now or later to outcome parameters. One thing we've learned is that the test can detect about 100 copies per ml or about 10 copies per reaction, and it's very specific, probably greater than 99.5% specific, but the virus doesn't like to read the textbook, so sometimes you can get false negatives early on in infection, and so the clinical sensitivity still needs to be discovered in a well-studied population. And the predictive value, of course, depends on the population that you study. Next slide. So in terms of genomics, what's going on there is Natalie, and you'll hear from her in a few moments. She leads the CDC team that collaborates with Richard Harrigan and the Snutch Labs from UBC. And this research is funded by Genome BC and CIHR, and 177 genomes have already been sequenced and many posted on GZAID. It's a slowly mutating virus. The sequence depth is of limited value, And the sequencing helps identify transmission into BC related to contained clusters, but also identifies the yet-to-contain outbreaks and local transmission. There are IT solutions to integrate these whole genome sequencing variant calls into the existing lab data, and really a question that we need to collectively answer is how best to use real-time WGS to guide how we respond to this outbreak. Next slide. In terms of serology, there's a plethora of unvalidated point-of-care and lab-based serological tests. There's an intense collaboration with the National Microbiology Laboratory, and fundamentally the questions are seropositive healthcare workers immune? There's some data from monkeys that say this is true, and it fits with other coronaviruses. What is the true infection denominator to help David answer the question about how many individuals in BC have actually gotten infected without having significant symptoms? I'm not convinced about the serology test for clinical care, but there's some evidence that it could help you resolve some of these cases where the NAT is negative. And again, as we roll out serology, there's issues about analytical sensitivity and specificity versus clinical sensitivity and specificity, and these remain to be characterized. Next slide. So fundamentally, what we have is provincial assays that tell you NAT, viral genomics, and serology, and what we already have seen is physical distancing reduces the R0, and it's a reduction tool. What has been the spread in BC are important questions. Who has already been infected? And there are a number of options how to assess that. That includes understanding the prevalence of active infections by NAT, serological assessments of prior infection in various populations, and the use of strategic genomics, especially when we begin to lift some of the restrictions. There's a lot of questions on how to do population level sampling, what's the statistics we're going to use, the modeling that we're going to use, and could you use targeted self-collection for COVID? Could you use serological surveys using pooled samples or dried blood spots? And that's all I have to say.
Hi there. And I'm Natalie Prashicki. I'm a microbiologist here at the BC CDC, and I co-lead the molecular microbial genomics program. And so that is our R&D section, where we were the ones to help develop the assay that we're using for diagnostics, and we're also working to develop methods to perform the sequencing and to understand its utility during this outbreak. So I really wanted to highlight how quickly we were able to access funding to be able to carry out this work. Our first positive sample occurred on January 27th, and we received our first influx of funding from Genome BC on January 28th. And likewise, the funding that came in through CIHR to supplement that work came through very quickly as well. So very thankful to those funders. And I also want to recognize that we have a large research team working on the genomics that is we're working together with Richard Harrigan, Terry Snatch, Mel Krashen, and the team here at the BC CDC. So it's really an effort of many people. Our sequencing efforts have really focused on one of the next-gen sequencing platforms called MinION. This is a portable sequencer that's really very suitable for working on viral sequences. We can multiplex up to 24 samples at a time. We have the capacity to do hundreds of samples per week, and we can generate near whole genome sequence data in almost real time. And so really the turnaround time from sample receipt to having sequence data to analyze is 24 hours. And so this allows us to be able to really envision how this method could help us with a rapid response, for example, in understanding the transmission or if there was a novel outbreak. If you go to the next slide. And so to date, we've sequenced 177 genomes, and as Mel mentioned, we've been releasing this data in close to real time to public repositories. So the one that we've been extending our data to is GISAID, which has been the place where majority of people who are sequencing genomes around the world have been depositing their data. It was traditionally used for influenza. And I think one of the things that's really interesting about this outbreak is the speed at which the sequencing has occurred and the number of groups that are sequencing. So if we look back to what happened with SARS, back in 2003, it was about 6 months before the first genome was released, and that was done by leaders here in British Columbia. In contrast, here with SARS-CoV-2, the first genome was released by a group in China within 2 weeks of being notified of the outbreak. And likewise, we've been sequencing since February. And so this means that there's lots and lots of genomes being released in real time, and they're predominantly being released to a program called NextStrain, which is aggregating the genomic data from around the world so we can start to really understand the transmission in near real time. And what you can see here is a slide with the BC data that has been released publicly so far, and you can see that there's certain groups that tend to cluster together or to form clades. And if we look at the closest relatives from afar, we can see that we have some clusters that have the highest similarity to samples that were initially identified in Washington State. And we also have some that are more similar to what we see in Europe. And what we can do with this is we can start to make those associations of where the travel may have occurred, or now that we have primarily community transmission, we can start to understand the transmission within our community. And so if you go to the next slide. So the value of this data is to understand the transmission, but its real value needs to be amplified when you start to integrate that with the epidemiological data. And so this is work that is funded. Our project is called Respond, and it's funded through the Canadian Institutes of Health Research. And what we intend to do with this work is to take the 2,000 of the genomes that we sequence and integrate them with the IDEAS cohort, which has risk data, so pre-existing conditions, as well as outcomes data. And we will integrate that in a data management tool called Clover that we're already using during this outbreak to be able to look at the integration of genomics data and clinical data in near real time to really understand the linkage and what this can tell us about the virus. Now, it's a nice study design, but it's not complete. There's still some missing data parts, and one is that we still need to work on refinement of the viral genomic methods, and we've been working with Terry Snetch at the University of British Columbia to do so. But we also but the other thing we're missing is the human genomics data to understand if there's certain human features that predispose someone to having a more severe outcome. And that's work that Stephen Jones will be presenting in the next upcoming slides. Thank you.
Stephen.
Can you hear me now?
Yeah.
Great. Okay. So I'm gonna briefly talk about what we're doing in terms of germline analysis of the host. So that in this case are the human patients. I'm co-director and head of bioinformatics at the Genome Sciences Center here in Vancouver. If we go to the next slide, please. In terms of our team, I'm not going to go through everyone's name here, but obviously we're drawing from people from the Genome Sciences Center, a lot of people from the CDC, BC Children's Hospital, St. Paul's, and SFU as well. So a great team of people coming from both the epidemiology, the clinical variates, the human genetics, and the technology aspects of this as well. So if we go to the next slide. What we're really trying to do, and we're not unique in the world in this, there's a lot of other groups around the world which I'll come to later who are trying to contribute to this goal as well, which is to understand the genetic factors that influence the trajectory of the disease, right? So we know that especially for people who are younger and have no comorbidities, many of those people get mild symptoms, but still many of those people get a severe infection. And what we really want to understand is there a genetic basis for that in part or in whole? And could we actually determine what those genetic biomarkers were so we could predict whether a person, for instance, is likely to have a more severe infection? Likewise, essentially identifying people who would be at unusually high risk of being infected and having a poor outcome. Certainly understanding the genetic interactions of how the virus interacts with the human body and the variations between individuals, how that affects the disease. certainly can give us some very fundamental insights into how this disease process takes place. And certainly understanding many of those pathways in terms of the response to the disease and how infection takes place can also give us therapeutic insights. And very important too, to something that we're very well grounded here in the GSC, and I think Canadians as a whole too, is that we also want to make sure that we can share our data with the global scientific community. If you go to the next slide, please. So what we hope to be doing is taking or getting the complete genomic sequence of the human host. We'd be wanting to get what we call a complete genome. So that is a genome that is done at approximately 30x redundancy or greater. We have the capacity at the Genome Sciences Center in terms of equipment and qualified personnel to do about 10,000 genomes per year. And what we've been able to show through trials that we've already done is that we can actually get the human genome derived from the residual test material that actually comes from the nasal swab itself. So we don't need to go to patients and get a blood sample, for instance. So that, I think, really expedites our ability to do this. There is the potential, conceptually as well, that from that same material we could derive a COVID-19 genome at the same time. But also what's really interesting as well is by analyzing the nasal swab, we're actually also going to be surveying the metagenome of the airway. So whether bacteria that you naturally have, which might be different to other people, in this airway, whether that actually affects the disease trajectory will be something that may also come out of this. If you go to the next slide. And this is just one example of the kind of things that we could find. This is a paper that we submitted earlier in the week. So the ACE2 gene, for instance, is the gene by which the virus actually attaches to the human cell and basically allows the infection. And there's certainly within the human population a lot of natural variants that exist. And so one of the things that we could easily start to ask from this study is, are some of these variants more permissive for disease? Are some of these variants, for instance, protective for disease? And just to point out too, not many people kind of mention this, but this ACE2 gene is actually on the X chromosome. So potentially too, we'd be able to look for slight differences in how the genetics behave for, say, males and females. And if you go to the next slide. Just to point out too, as I alluded to at the beginning, there are groups all over the world now trying to do this, right? Because we're really going to have numbers of human genomes in patients and understanding the clinical parameters that they exhibited to actually make sense of this data. So there's an international consortium, the HostGenix Initiative. Many kind of world-renowned famous groups participate. The Wellcome Trust Sanger Institute, the Broad Institute, for instance, and kind of many more on this map that you can see. So really what we're trying to do is, be part of Canada's contribution to our understanding of how humans interact with this virus. And I believe that's my last slide. So thanks everyone for their attention.
Thanks very much, Steve. Given our time, folks, I think we'll go straight to Dr. Unrau's discussion on diagnostics.
Okay, I think I'm unmuted now. All right. So when we heard about the pandemic starting to emerge, we filed a grant to CIHR to try to develop a rapid detection test kit. This would have the advantage of being able to be easily reprogrammable to other viruses in the future, and it would also be potentially possible to distribute this kit far more diversely throughout British Columbia so that other people can easily implement this kit with minimal technological training. So we put together a team of RNA biochemists. The RNA biochemists are largely in this photograph. organic chemists, virologists, clinicians, and some engineers to try to make that possible. So if I could have the next slide. Of course, we're not all standing around like in the last slide. This is now the correct spacing. And if we could have the next slide, please. So what a key technology that we've been developing is RNA imaging tools. Our original focus has been inside of living cells. And so what you're seeing on the upper left there is a paper that just recently came out in Nature Chemistry, or Communications. But we have these RNA aptamers that can bind a fluorogenic dye, which is in the upper right, and become intensely fluorescent. So they're brighter than enhanced GFP. And so we reasoned that we could use such aptamers for detection by synthesis. So rather than using a chemical fluor that is always fluorescent and then reports by a variety of more complicated strategies the detection of a nucleic acid, you actually build the detector out of RNA, and then it binds the fluorophore, which is intrinsically not fluorescent until it binds to the RNA that it is targeted to. So the RNA aptamer and this fluorophore bind very tightly. So once that interaction happens, you turn on and you can see things inside living cells, but we thought we could use this also just as an RNA detection tool. So if I could have the next slide, please. So we developed an isothermal system based on an existing NASBA technology. This is an isothermal technology. It's primer-based. It's a lot like PCR in spirit. So it uses pairs of primers, but it uses 3 enzymes, so T7, reverse transcriptase, and RNase H, to isothermally amplify an RNA. So if you look at the outer reaction, it would just produce an RNA that you could not see, but the inner reaction has been modified with that a green handle. That is actually just encoding for the mango RNA to be produced. So by nesting, we found we could detect as little as about 1 molecule per microliter, which is very close to the sensitivity of what we have with the testing that is now going on for clinical samples. If I could have the next slide. So what we're hoping is that we can optimize primers against the virus, that we can design a small isothermal unit that can actually detect the fluorescence. And so what you're seeing in this little movie here, sped up, so this is over actually about 11 minutes, is the emergence of a fluorescent signal, right? So you can actually just see that with your eye. So this little unit that we're thinking about here, you could just look in through the top and see if you have a positive. It would also have electronics that would quantitate how much fluorescence you have and allow that to be uploaded and reported to some central service. So what we're hoping to do over the CIH response research is to develop such a kit, simplify considerably the collection of the sample, which is still something that has to be done, from saliva, we're thinking, and in principle make something that is much easier to implement. You would still need a technician of with some scientific training to implement such a technology, but it could be distributed much more diversely than is currently the case. If I could have the next slide, please. Yeah, so some gaps, I think. Really, what is the simplest way to preserve enzymatic function in saliva? We're working on that with a team that I've collaborated with at Imperial College in London. What really is the sensitivity that you need? As low as possible, obviously, but if you go below 1 molecule in the tube, you're just not going to get anywhere. And really, the ultimate question is for reliable testing is, can you actually get the testing to work at home? So that, I think, is a very complicated question to answer. It really has 3 problems. How do you combine simple detection, stable enzymology, and usability such that somebody could use it rather like a pregnancy kit is used today? Some other questions I think that we've come across is, what is the viral load in asymmetric carriers? I think maybe some people in this audience actually know, but we don't know the answer to that. And how does viral load in saliva and swabs actually compare to each other? And we've noticed perhaps some higher level issues just with coordination over governmental levels of emergency response and institutional levels of emergency response that could be coordinated as a result better as a result of this particular pandemic. So if I could have the next slide. Yeah, so what our team has to offer is that we have samples, saliva samples being collected at St. Paul's and with collaboration with Don Sin and Mark Romney, who are doing RT-PCR analysis on swabs of those patients. We have RNA biochemistry expertise and chemical synthesis expertise to offer, viral culturing and testing through Massa's laboratory. Really have no administration capacity whatsoever, but hopefully we'll get some more in the near future. Next slide. I'd just like to acknowledge all the people that have made this project happen so far. And with that, I'll end. Thanks.
Thank you very much, Peter. We are a little bit behind, so I'm going to turn the mic over to Rob McMaster for the next session.
Thank you, David, and really thanks to the previous speakers. As you can see, you know, everyone is working in these really productive teams, which you need to in a pandemic. So we're moving now on to hear about clinical trials and patient management. We have 4 speakers, and listening to them, a few of them earlier today, I think all 4 are actually in clinical trials. They're certainly ramping up for clinical trials imminently. So the first one is Josef Penninger, the Director of Life Science at UBC. And Josef is calling in from Austria, where he is spending his time with the travel ban on moving between continents and between countries. So Josef, thank you.
Hear me? Can everybody hear me?
Now you're coming in.
Okay.
So good evening to Josef.
Yeah, hello. It's 11 o'clock.
Yeah.
I come to you from Vienna. I'm not allowed back to Canada because of the travel ban. I'm not a Canadian citizen. And so I want to tell you, I will be very fast. 20 years of work in 5 minutes about ACE2 and COVID-19. Okay. So, okay. So in around 1999, we actually cloned ACE2. A fellow I had when I was in Toronto, Mike Krakauer. We cloned it from a screen from Drosophila, and we made the first knockout mouse. And at the end of the day, this is the sum of many, many papers of my lab and many other laboratories. ACE2 is the negative regulator of the renin-angiotensin system. It basically inactivates angiotensin 2. By doing so, is a tissue protective factor for multiple tissues. The heart, the kidney, the liver, and also what we found is also a protecting molecule for the lung. How do I change this? Sorry. Okay. And then it turned out that ACE2 is actually is involved in lung injury and lung edema. So basically, if there are different types of injuries, and we developed very early on, so the knockout mouse we published 2002, and then the connection of ACE2 to the SARS virus and to lung injury we published in 2005. So I'm into this for a long, long time now. And we developed lung failure models at this time. So if we have lung failure models, unspecific lung failure models like acid aspiration, sepsis, the renin-angiotensin system is actually induced, and via the AT1 receptor drives a lung edema, opening of blood vessels, leaking blood vessels, leading to much more severe acute lung failure. In this system, again, ACE2 is a good guy. So if we actually take away ACE2 from the system, then the lung injury and the lung failure becomes much more dominant. So we mapped this all genetically of ACE2 knockout mice, ACE knockout mice, knocked out all the receptors. And you're probably aware of this raging debate at the moment if ACE inhibitors could be useful or AT1 receptor blockers, ARBs. So this basically goes back to our paper, which we published in 2005 in Nature. So then it turned out, initially proposed by a group in Boston, 2003, that the SARS-CoV coronavirus spike protein would use ACE2 for entering the tissues. There were other receptors out there which were proposed like L-sign. So we did the first in vivo experiment because we had the first and the only ACE2 knockout mouse at this time, so we sent it to China. We infected it with SARS coronavirus, and the bottom line was without ACE2, there's no infection. So this was basically the critical experiment to show in vivo that ACE2 is the essential receptor for the SARS virus, no ACE2, no infection, which does not, of course, exclude cofactors and co-receptors, but ACE2 is absolutely the critical receptor for this. So we also figured out at this time, or we proposed one of the mechanisms by the SARS virus became a killer virus, and actually we believe that's exactly the same now for COVID-19. Because the SARS virus became a killer virus because it actually binds to ACE2, and this is the entry gate into the cell, but also the virus takes ACE2 with it and leads to ACE2 downregulation. So very similar to HIV, that the virus binds to CD4, takes away CD4, and people lose immunity. This viruses, SARS virus 1 and the SARS virus 2, basically bind to ACE2, take it away from the surface, and by doing so, they enter the cell, and secondly, ACE2 is tissue protective, and this is why we believe that the severe disease, because you lose expression of ACE2. So this is what we had figured out many years ago. The next slide, please. And based on this, we developed a recombinant human soluble ACE2 molecule. This was one of our experiments where we figured lung failure, and when we give the drug during the lung failure, we can actually alleviate, make it a little better if we provide an enzyme therapy for lung injury, because we and many other people have shown lung injury also triggers the renin-angiotensin system. ACE2 is downregulated, so we're basically supplanting the enzyme. So the next slide. And of course, then the new virus came along. So we have been working on this for basically 20 years. As I said, we had cloned ACE2. We made the first knockout mouse. We had this first connection to lung injury, lung failure, the first in vivo SARS experiment. And of course, nobody cared anymore because the SARS virus had disappeared. And this new virus appeared on the sequence. When the sequence was published, it was quite clear very fast that ACE2 should be also the receptor for this virus. So the next slide, please. And since we had the drug in development, actually since more than 10 years, so this human recombinant soluble ACE2 molecule, so this molecule went into phase 1 clinical testing in healthy volunteers via a biotech in Europe, which I had co-founded many years ago. And it also went into phase 2 clinical trial with people with lung disease, so pulmonary hypertension, and also some ARDS, acute respiratory distress syndrome. So all in all, 89 people already received this molecule, which were put into clinical development. So this is the molecule we call APN-01, recombinant soluble human ACE2. We had the virus in Sweden together with Ali Mirasimi, so I do actually my virus work at the moment on COVID-19 causing coronavirus. In Stockholm, there's a high security lab, so I work with Ali since many years on Ebola infection, Marburg virus infection, Lassa virus, we call it. And this was the experiment when we infect the Vero cells with the virus and give different doses of recombinant ACE2 and also different doses of the virus. You can see in a very nice dose-dependent fashion, human recombinant ACE2, APN-01. This is exactly the same molecule which actually will go now in clinical trial. Reduces the virus load by 1,000 to 5,000 times. So we are doing this now actually side by side with remdesivir, with chloroquine, and some other drugs which are out there and have been claimed to work. Some of them in our assay do nothing, but we are repeating the experiment. So I don't want to make claims yet without having at least repeated it 2-3 times. So here also, if we actually use mouse ACE2 to block, it does not work because the virus has no affinity for mouse ACE2. Good. The next slide, please. The other thing we did, we engineered human blood vessels, something we had published last year. This is how they look. And indeed, here in B, you can see the virus can grow in these human blood vessels. We know now ACE2 is expressed in pericytes and also in endothelial cells. So this was, I think, a very important experiment, because if the virus spreads in the body, it's actually because of size constraints, has to infect the blood vessel to get into the tissue. So in C, you actually see we can get back virus which infects other cells. And in D, if we give recombinant ACE2, we can significantly reduce the virus infection and the viral load in this blood vessel organoid. So engineered human tissue, which we believe could be relevant for drug testing, for studying mechanisms of disease, for testing some vaccines in the future. So the next slide, please. We did the same together with Nuria Montserrat in Barcelona. She developed these organoids, these kidney organoids. They're very special because they've proximal tubules. This is where ACE2 is expressed. So ACE2 is not only expressed in the lung, it's expressed in multiple tissues. And this is why we, and of course many others, believe that the virus spreads into the kidney, into the heart, into the intestine, into other tissues like the heart. And I think one point I would really like to make, and you know it anyway, COVID-19 is not a lung disease. COVID-19 is a multi-organ disease. And it also has massive influence of the blood vessels. There's this coagulation deficiency, there's this involvement of cardiovascular systems. So this is a multi-organ failure, not just a lung disease, at least the severe cases. So again, the virus grows in the kidney organoids. In B, in C, you can see we can get the virus back, which can infect other cells. And here, actually, this was not in our paper, which we published last week. These gut organoids we got from Stem Cell Technologies in Vancouver, and we can also infect them. And as we speak, we're also doing now lung organoids to infect them with the virus. And I can tell you the virus also grows very well. And of course, now we're doing all these experiments one has to do with, we combine this now with single-cell sequencing, single-cell transcriptomics, what actually does the virus do to the cells which are not infected. We set up all immunohistochemistry to see which cells are being infected. And of course, the idea is to provide human tissue organoids to study the virus in a relevant human tissue. And of course, one can test the drugs on them and see how the effect is, or test in the future vaccines on this. So the last slide. So, and based on all of this, this molecule, APN-01, which is recombinant soluble ACE2, it has actually two functions engineered in the same molecule, because from the structure, they're different in different domains of the molecule. One is actually reducing the virus infections I've shown you. It makes sense. It basically acts like a neutralizing antibody. It has very high affinity. I think the affinity is 1.6 nanomolar. So this is like an antibody binding an antigen, ACE2 binding to the spike glycoprotein. And secondly, it's protecting multiple tissues from the damage of hyperactivation of the angiotensin II system. For instance, if we inject actually humans, and there was a human trial to my amazement, inject them with angiotensin II, and Ang2 is actually shooting up in COVID-2 patients and correlates with the severity of disease. If you inject Ang2 into humans, the biggest side effect is thromboembolism, which is very interesting. And the other side effect is actually it induces heavily a cytokine storm, in particular interleukin-6. So of course, there are many more mechanisms which cause the disease, but we think that's one of the critical mechanisms. So it's in the same molecule. So this protection of organs is actually depending on the catalytic domain. It's a function of cutting angiotensin II, inactivating the system, and reducing the viral infection is of a different domain, and these two functions are independent. This molecule was now approved for phase 2B clinical trials in Europe, in Austria, in Germany, and in Denmark. And maybe starting from tomorrow or early next week, the first patients will be recruited. The clinical study is placebo-controlled, double-blinded. 200 patients will be recruited in 11 clinical centers. In Germany, it's Hamburg and Munich, the University of Munich, 4 universities in Austria, and all the hospitals of the University of Copenhagen participating. The trial will be 7-day infusion of recombinant ACE2 IV twice a day because of the half-life of the molecule, which is around 12 hours, and then every patient will be followed up for 3 weeks. But since it's double-blinded, we can only know what's the outcome after the last patients are recruited and followed up for 30 days. So we hope in early summer we can unblind the trial and have a clean conclusion. It works or it does not work. And of course, there were many clinicians and many clinicians involved. Canadian clinicians from Toronto mostly, from Copenhagen, from Germany, from Austria, which developed the trial protocol. And the treatment will be in people with severe COVID-19, so the third stage of COVID-19, with the clinical outcome that we stabilize them and they don't progress to the final stage of the disease. And with this, thanks for listening.
Thank you, Josef. I think we're running a little short on time, so move right into Srin.
Hello, can you hear me?
Yep, thank you.
Great. So I'm Tony Rasmurthy. I'm an intensivist at Children's Hospital here in Vancouver. I'll be talking about the CAPCO trial, which is in conjunction with the World Health Organization's Solidarity trial. We have a Twitter account, an email account, and a logo. We're very exciting. Next slide, and I'll spend most of my time on this slide. This is a global mega trial, we're calling it, with the aim of enrolling thousands of patients around the world with a common analytic data set. It's a registered trial of 4 separate interventions, antivirals for all hospitalized adults with confirmed COVID-19, remdesivir, hydroxychloroquine, Kaletra versus optimized supportive care, with the primary outcome of hospital-level mortality. Sample size is impossible to calculate because this is an adaptive trial. We will be incorporating new interventions as they become available into this randomization scheme and dropping interventions from that list as they prove to be harmful or futile. There is a global DSMB, which will be constantly monitoring the accrued data set to see if any of those signals are present, effectively being one randomized trial for the planet. Currently, it's in approval stages in 83 countries around the world, with its chief investigator, I guess, technically being Dr. Tedros. Obviously, this is a complex and innovative approach to clinical research. The WHO is not a research sponsor in the classic sense of the word, but they felt, and I think the world and the ministries of health around the world felt, that this was a unique circumstance that required a unique approach. And so the principle here is that with more patients being enrolled in one data set rather than small, differently controlled and differently performed trials with different outcome measures, the ultimate meta-analysis that would come at the very end of all of those trials reaching their primary outcome would take too long, and this is the approach that's been proposed. Currently in Canada, we have funding from CIHR to do it in about 25 sites, and we're actively seeking out other envelopes of funding to expand from there. We have at least various stages of approvals in 85 sites across the country. We do not have funding for all of those sites, and basically we're walking around with our hats in our hands to all levels of government to see if they're willing to support this further. The drug will be supplied through the trial, through the WHO or through relationships that we've made with the manufacturers of each of those drugs. And it's a very straightforward, simple design of confirmed COVID-19 inclusion criteria, a small set of exclusion criteria, and a primary outcome of hospital mortality. Reports will come out as data is available, primarily through the WHO, linked to the public health agencies of the world, and so not the classic publication-based reporting as with usual studies. And the global DSMB has the ability to report directly to Dr. Tedros. Papers will be all-inclusive, and so we call this the Higgs boson paper of clinical research. There will likely be 5,000 to 10,000 authors, all as co-authors of the manuscripts, given the numbers of sites that are participating. So obviously it's a complex initiative. In British Columbia, we are starting in a handful of hospitals in the Lower Mainland and on the island, with hopes for fundings to expand out, acknowledging that access to these drugs is extraordinarily limited, and ideally, and it is only through the context of a clinical trial, which is the current guidelines both from the Public Health Agency of Canada and the BC CDC. And so we have an obligation, arguably, to include as many patients as possible within the context of this clinical trial. And so ongoing funding support is being sought. Next slide. As part of this grant, we are also contributing to a global data set of COVID-19, which is housed in Europe and through the World Health Organization once again. This is a preliminary reports, for example, and I just put up a couple of targeted data sets that have already been released and informed public health. Currently, there's approximately 8,000 patients in this global data set, and about 45 to 50 hospitals in Canada are contributing data currently. It's a very straightforward data set for analysis with a 3-page case report form that produces reports globally that go directly to the World Health Organization for both research and operational purposes. And this is a figure of days relative to admission and outcome status, looking at discharge rates, death rates, and the timing therein. Obviously, from a public health planning perspective, this is very useful data. Next slide. Another similar figure looking at, say, location in hospital and length of stay. For reference, Canadian data, we have this is global data, but we have the R code available for participating sites to do their own analyses of this accruing data as it continues to accumulate across Canada and across the world. Next slide. Also from CIHR, we were funded to do some ancillary work with the WHO once again to do a qualitative evaluation on healthcare worker comfort and familiarity with PPE and working during a pandemic, and that is ongoing in conjunction with colleagues in sub-Saharan Africa, Hong Kong, China, the EU, and other places in Southeast Asia. Get comparative analyses of healthcare worker comfort with managing a pandemic and relationships to PPE and familiarity. Also, surveys on clinical researcher comfort with research during outbreaks, and that manuscript is currently in submission. And that is all I have to say within my 5 minutes of a lot of work.
It was a huge amount of work.
Obviously, a large global team working together.
That's great. Thank you, Surin. We'll just move straight on to Jim.
Okay, can you hear me now?
Yep.
Hi, good afternoon, everyone. My name is Jim Russell. I'm a professor of medicine at UBC and a principal investigator at the Center for Heart-Lung Innovation, and I'm pleased to speak to you today on behalf of what we call the ARBS Corona investigators. The study is closely aligned with the studies that Srin talked about. We've been lucky enough to be sponsored with a grant from the CIHR, and we've also been supported by generous donors sending funds to the St. Paul's Hospital Foundation and some of my own research funding. So next slide, please. So the rationale for our study builds on some of the work that Dr. Pettinger just talked about, and that is that the ACE2 that you've heard about is the receptor, the primary receptor for COVID-19, the SARS-CoV-2. And in previous studies of viruses that also bind to ACE2, such as H1N1, it's been shown that angiotensin receptor blockers, or ARBs, the oldest one is called losartan, worked in murine models of lung injury to reduce both viral titer and lung injury, very similar to the rationale that Dr. Pettinger talked about with the recombinant ACE2. And the results of that very preliminary work, surprisingly no human data prior to writing this grant on the effect of ARBs in prior influenzas that use ACE2, we came to the hypothesis that ARBs could improve outcomes such as need for ventilation and death in adults hospitalized with COVID-19. And the figure shows, for example, the ACE2 deficiency, the little blue circle at the top, not acting enough to deregulate and take away the effects of angiotensin II, a potent vasoconstrictor, which normally binds to the receptors, particularly the angiotensin II type 1 receptor, leading to the multi-system organ failure, not just lung injury. And that the ARBs can block that effect. They're very effective. They're used in 30 to 60% of patients with hypertension and heart failure. And so that could mitigate the effects on organ injury. Next slide, please. Secondary hypothesis that we're working on in this study as we collect patients is to look at predictive biomarkers. And predictive biomarkers, by definition, means biomarkers that can identify those patients who respond better. That means better safety, better efficacy. The candidate biomarkers that we're starting with are plasma angiotensin 1 and 2 levels and other markers in the angiotensin pathway, as well as panels of proteomics, metabolomics, and inflammation. Next slide, please. We're particularly interested in acute cardiac injury for a couple of reasons. First, patients with heart disease, hypertension, diabetes, and chronic kidney disease have high risk of getting COVID-19. And secondly, as I said, 30 to 60% of those patients are on ARBs or ACE inhibitors. And then third, fully 40% of patients who are critically ill with COVID-19 get what's called acute cardiac injury. The pathophysiology shown simply here at the top, the virus itself can infect cardiac cells, as we've heard, and cause injury. Diseases such as hypertension, heart failure, diabetes can act through angiotensin II, causing vasoconstriction and ischemia, shown on the lower right. In fact, there are now case reports of patients with myocardial ischemia and infarction with normal coronary arteries. And there can also be myopericarditis, also case reports in circulation recently. These conspire to produce acute cardiac injury, and when that occurs, there's a higher risk of multi-system organ failure and death. So the notion, very similar to what Dr. Pettinger said, is that the virus starts in the lung, but then spreads to these other organs, including the heart, kidneys, intestine. Next slide, please. And these ARBs, such as losartan and cosartan, interestingly have been shown in other studies, not particularly in COVID-19 or SARS-CoV-2, to block all of these different mechanisms. So there's a rationale and a putative biological mechanism for therapeutic intervention here. Next slide, please. Our design, it's a sample size of 500 patients. It's a pan-Canadian observational cohort in 25 to 30 centers in Canada. And we're including COVID-19 positive patients who are then either already ARBS exposed or not, and comparing them in adjusted analyses. Primary outcome, as we said, is ICU admission, ventilation, or death, but we're also very focused on safety because of recent questions about possible safety issues. Next slide. Potential impact, of course, since ARBs are readily available and have approvals for many conditions, could they be used to limit organ dysfunction mortality? We, in fact, would plan to seamlessly roll from this observational cohort to future trials based on the data herein. But if, on the other hand, they're proved to be harmful, then we believe that evidence might be useful for clinicians to guide whether or not to continue ARBs in patients with those conditions who are at risk for or develop COVID-19. And my last slide is a thank you for the groups that have put this together, other co-investigators, funders, the project manager, Jen Rocheleau at St. Paul's, and the 50 or 100 co-investigators, coordinators, and technicians across Canada. Thanks for your attention.
Good. Thank you, Jim. I think we'll just keep moving on. The next one's Rich Lester.
So, hi, greetings and thank you. So, Rich Lester, I'm an infectious disease physician at Vancouver General Hospital, and we were funded through the CIHR first round rapid COVID for our work in digital health to support case contact management in Canada and global outbreaks. Next slide. And first, my disclosures. In terms of being a physician and public funding, I'm also co-founder of WellTel, which is has a nonprofit and a regular company that provides the digital interventions that are informed by our research. Next slide. And our research team for this particular project is in conjunction with Dr. Giuseppe Carnini in data science at UBC, as well as investigators like Carmen Logi at the University of Toronto, social work, a world expert in stigma, and a number of clinical investigators throughout BC and the world, including in Canada, the UK, Rwanda, and Kenya, where the focus of this grant, as well as our industry partner, WellTel. Next slide. So our key objectives are really to deploy. This is already a ready-to-go service in the world in many of these countries, so that's why we decided to deploy there first. It was adapted to COVID quite quickly in January. But we're also doing some evidence review funded with this funding, rapid review, and then the latter two are really data science oriented, which I'll get to later. Next slide. So the rapid evidence review we've done so far is looking at digital interventions that may support COVID in the pandemic. So first we looked at tools for patients, decision support, symptom checkers, very popular as you know. However, there's a lot of evidence in randomized trials, and these have generally all been quite negative to inform patient care and decision making in the prior research. Tools for clinician on decision support. Quite a number of trials there too. There's several that do show some benefit in decision support, several that have null, and some show harm, which I'll show you. And then patient engagement in care, also a lot of evidence on what works, what doesn't, benefit and null. And pandemic support, of course, is more limited, which we can bring forward from previous experience. Next slide, please. In terms of do we need evidence for digital and digital support? So there's a lot of randomized trials, a lot of evidence out there. The largest, most recent AI and decision support randomized trial published in the Lancet Journal last year, actually over 65,000 deliveries that was done in maternal child health, actually caused an increased neonatal death of twofold odds ratio. So I think the answer is yes, just like with other therapeutics and diagnostics, we need to understand the impact of the interventions we're doing, including digital and virtual care. Next slide. In terms of, we were actually fortunate to be involved with the first trial using text messaging and mobile phone in the early pandemic in HIV to support patient care and behavior change. And we really worked out a lot of the details on how to text and best do that for behavior change. These have been validated through meta-analysis and various research in chronic disease throughout the world. And it's really the interactive nature and patient direct care that leads to behavior change. We've done some work there. Next slide. Suffice it to say, we've done a lot of research through the years, clinical trials in various conditions around the world, and written about that.
and trying to inform. So in terms of the public, yeah, next slide, please. In terms of our number one for COVID is public health support. So we were able to adopt the WellTel program to support patients on public on home isolation and assist with case contact management real time. So someone will get enrolled who's expected to be on home isolation, get daily text message check-ins. We know that has the greatest reach for 2 weeks during their incubation period. That's followed up by public health nurses, about 100 times more efficient than phone calls. And Rwanda adopted this a few weeks ago rapidly, and their entire nation COVID response is enrolled in the WellTel program. Cases and contacts, about 100 to 1 contacts per case. And you can see they may even be bending their curve at below 100 cases, despite having about 3 or 4 times the population that we do, compared to the curve on the right in BC, which is much higher and much steeper. You can Google some stuff about this. Next slide. In terms of the other thing is just virtual care, which was the original application. We've done work to support asthma care, chronic disease, primary care in the north, which has been a benefit. We started work in mental health and hospital readmissions. So of course that work's ongoing as everyone goes virtual, just as this symposium has done. Next slide. And then really what a lot of the research science on our data is really looking at this. We have an incredible opportunity to analyze natural language between patients or public and care providers. Because we use an open language concept, you actually get better data than if you send pre-described forms and so on. Open data, we can analyze that with natural language processing into predetermined topics like diagnostics, treatments, even lifestyle and social. And then now we're adapting for COVID-specific language and even drivers of stigma, perhaps. We'll develop something that looks at that. So, so we really will learn a lot from the patient perspective on what their priorities are, how they're affected. And we actually have done some prior predictive analytics in HIV, and we can actually, using metadata, predict whether someone's going to have a good health outcome or not well in advance of other means. Next slide, please. So my take-home points are there is an evidence base for virtual care, and we really ought to know it and use it to guide in the pandemic. There's a lot of knowledge there. We need to deploy fast and be in pandemic mode. We should be ahead of the curve as wherever we can rather than chasing it. And we need to learn. We need systematic research to keep informing where we are now and in the future. Back to David Patrick's sort of analogies, innovation wins wars. We got to keep innovating, but also trust wins pandemic. So it should be data-driven with transparency. Thank you very much. Next slide. Everything virtual. Yeah.
Thanks, Rich. I think we've actually exceeded our time for this session, so we'll save questions for later. But I do notice now, because we actually have people waiting to come in. We've maxed at 500. We now have a streaming, and if you look in chat, there's a message where you can actually stream live rather than coming in through Zoom. And that will enable either more people to come in, read the message, and then start streaming. So we should be able to now attract everybody who's waiting if the message gets out. Thank you for the speakers. I think I learned a lot from this session on some of the complications from COVID, that it's not just a lung disease and the mechanisms that's occurring. So with that, we'll move on to epidemiology and public health response.
I agree. It was a terrific session. So we're on to 3 5-minute talks on epidemiology and public health response. And we will have time for questions if the speakers can finish in 3 or 4 minutes, but otherwise we'll keep carrying on as we're carrying on. So first up will be Dr. Danuta Skavronsky from the BC Centre for Disease Control. And she'll be followed by Manisha Durangani from BC Children's Hospital, and then the dynamic duo of Caroline Cullen and Michael Otterstatter from SFU and CDC, respectively. Danuta, are you muted?
Okay, I should be unmuted now by the host. Can you hear me?
Yeah.
Okay, excellent. Okay, so hi everybody. I'm, as David said, an epidemiologist at the BC Centre for Disease Control, and though many of the features of COVID-19 have been worked out in the weeks or months since we first learned of this emerging pathogen, there are still some very basic epidemiological characteristics that remain poorly understood. And I just want to share a couple of those that have been preoccupying me and I think many others in recent weeks. So next slide. Chief amongst these, as basic as it may seem, is what proportion of the population actually has been infected. And although there are some elegant approaches to try to compensate for the fact that we know we are not capturing all of the accumulating cases in the population, even those methods are also dependent upon some stability, at least in the testing indication or the eligibility for testing. But as those testing indications have expanded and contracted, it's really confounded our ability to get a clear sense of what proportion of the population has been infected. And of course, that's very important for us if we're imagining a time when we may have accumulated sufficient immunity, perhaps, to alter the course of the epidemic. Next slide. And of course, chief amongst that consideration related to the proportion infected is the importance of mild or asymptomatic infections. And there are a number of papers that have been attempting to quantify that, demonstrating that when there is enhanced case detection, active contact-based or household-based follow-up of contacts, we do uncover a substantial proportion of cases that are, if not asymptomatic, at least pre-symptomatic. And recognizing that children may more often fall within that milder spectrum of disease, also uncovering when actively assessed a comparable rate of infection amongst younger individuals, including children. So in this study, about 20% overall asymptomatic. There are a number of other studies that have shown as much as 50% of cases may be asymptomatic, at least at the time of virological detection. That's really important for us to get a handle on, again, if we want to understand evolution in population immunity. Next slide. And if we do accept that the proportion of clinically evident cases increases with age, as shown in this elegant analysis, if you go to the next slide. Well, then this also confuses our understanding of what is the actual age distribution. These are plots that were prepared by David Roth here at BC CDC, because incredibly, if you go to the WHO website, in fact, you will not find age distribution of cases. We have over 1 million cases globally now, and I don't suppose they have it on all of those cases, but not even amongst the subset do you find that basic information, descriptive information of the age distribution. So David has culled this information from various websites, and what I want to point out is how varying the age distribution is. I think a lot of people have seen the comparison between Iceland, for instance, where testing eligibility is very liberal. They offer tests to anyone who is symptomatic and wants it, compared to the Netherlands, where it is far more targeted. And you can see that when you test more, you actually uncover cases in that younger extreme of age compared to the older age group that is otherwise dominating our surveillance information. You can see where British Columbia sits. We, relative to the rest of Canada, let alone compared to Iceland, are likely missing children, school-age children in that 10 to 19-year age band in particular. So without a good way to assess true infections in the population, we're also skewing our age-based risk assessment. Next slide. And finally, if you want to be able to quantify what is the risk or the likelihood of serious outcomes overall or by age group, again, you need to know what is that true underlying case count or denominator. Go to the next slide. So we have previously, our team used seroepidemiological or immunoepidemiological methods many times around other emerging respiratory pathogens, including during our avian influenza H7N3 outbreak in 2004, with H3N2 swine origin, H3N2 variant in 2012 thereabouts. Most recently, 2015, with the importation of our H7N9 cases, we did a snapshot to assess population susceptibility, and also during the 2009 pandemic. And at that time, we were trying to understand why the very old seemed to be preloaded with some kind of pre-existing protection even before the pandemic, that rendered them less likely to become cases with the lowest incidence in that age group, highest incidence in school-age children. And we were quite surprised when we looked at sera that we had collected pre-pandemic to find that the very old, notwithstanding this being a novel respiratory pathogen, the very old had very high levels, nearly 90% of those over 90 years of age had a protective antibody against that H1N1 pandemic strain. And we weren't only looking at antibody, we were also looking at cell-mediated responses. So although the very old had pre-existing protection, if they became infected, we saw that they had the lowest cell-mediated responses to the infection. All that is to say how very revealing that those seroepidemiological studies were during the pandemic. And so when there was the COVID-19 emergence, obviously we were immediately thinking about these seroepi studies. Next slide. And just to show again how valuable serial snapshots, serial collections of sera and testing for antibody can be. Here you can see the pre-pandemic seroprotective profile for the H1N1 pandemic strain in 2009, and then post-pandemic comparing those seroprotective levels, very high levels now in the very young, reflecting their highest infection rates through the pandemic. And from that we were able, as I say, to derive the infection rates and contribute to global estimates of attack rates through the pandemic. Okay, next slide. So as I say, we very early on in the COVID-19 epidemic were considering doing these seroprevalence surveys. Unlike influenza, we don't have a protective threshold of antibody level to COVID-19, and there are going to be some nuances, obviously, to the interpretation of the antibodies that we find. But comparing to a baseline, this can be very informative. So the goal is to try to uncover that true infection rate, including asymptomatic infections overall and by age. We have collaborators in the Vaccine Evaluation Center, obviously at BC CDC. Life Labs has been really wonderful in collaborating with us, but we also have a serosurvey consortium nationally that we are linked with, and globally through the World Health Organization through what's called the Unity Study, which is a group that an international group of us who are conducting these seroprevalence surveys. So this is a cross-sectional serosurvey using anonymized residual sera. We'll collect 4 snapshots of 1,000 sera each. This is really just replicating what we did during the 2009 pandemic, 100 per age band across the lifespan, and not anticipating that we're going to have anywhere near 50% seroprevalence. This should give us reasonable precision within about 5%, I would expect, in those age bands. We want to assess by microneutralization not only ELISA-based for SARS-CoV-2, because of the importance of interpreting what those antibodies actually mean in terms of potentially interfering with viral replication. And we want to look at cross-reactivity to other humanized coronaviruses, notably OC43 and L63, and expecting that there would be a higher proportion of the very old with antibody to those other humanized coronaviruses. It would be important for us to understand what role those play in the potentially underlying immunopathology, as well as potential cross-protection. We want to collect baseline sera. We've already done that, actually. We've collected 1,000 already. We collected those. We were very fortunate to have collaborated with Life Labs very early on, and we collected the first 1,000 sera between March 5th and March 12th, which was just before the period that we thought we might be starting to see community spread, at least detectable community spread. I think that's very important because obviously we're intrigued by the relative flattening potentially of our epidemic curve here, and it would be very interesting to know then in that early March period to what extent population immunity that may have been accrued through potentially missed infections may be contributing. So I'm really interested that we move quickly to assess that baseline seroprevalence. But we also have 3 further serial snapshots that we are funded for, and the funding has come through the Public Health Agency of Canada as well as Michael Smith Foundation. And in the future, through this National Sero Survey Consortium, we are planning targeted prospective sero surveys of particular groups of interest, such as healthcare workers, remote and isolated communities, and First Nations with their collaboration. Next slide. Before we can start running those seroprevalence or the serological analyses, we need to validate them. And so obviously we're working very closely with Mel in his lab and also with the National Microbiology Lab to try to validate the plethora, the huge number of commercial assays that are available, and also to validate our own neutralization assay. So we have a second study, this immunity study, where we will be collecting sera from confirmed cases for the purpose of assay validation, but also to get a more detailed profiling of the immune response. So this is a case-based prospective study of confirmed COVID infection. We'll be collecting sera, PBMCs, and RNA at days 14 and 30 after illness onset. This is a collaboration also with Manish Savarandani at the Vaccine Evaluation Center. We propose 50 community-based virologically PCR-confirmed cases. We'll be looking at the type, the quality, the quantity, and the kinetics of the immune response based on those specimens. And we are currently recruiting via the Vaccine Evaluation Center. So for this audience, if you are aware of confirmed cases within 2 weeks of illness onset, if you could jot down this phone number and this email address, please provide that contact information and encourage them, if they're interested, to participate. We are otherwise drawing from our own registry of notifiable diseases and cases that have been reported to us, but it is extremely challenging, obviously, to capture these people within that 2-week period. We envision that the PBMCs and the RNA that we're collecting will be banking that to support other researchers as well that can contribute to the identification of antibody targets for supporting monoclonal and vaccine antibody and vaccine design. And we anticipate also that we could compare those profiles with other inpatient findings or those experiencing serious outcomes, these cases coming from the outpatient setting. And finally, next slide. We're also currently funded by the Public Health Agency of Canada to undertake a household study. Initially, we were predicating this on the WHO household transmission study, but recognizing we need a large sample size to achieve our main objective, which, as I say now, we are preoccupied with uncovering that asymptomatic fraction of cases. We are mostly focusing on the goal of measuring the secondary household attack rates, including the symptomatic and asymptomatic, whereas the WHO had also envisioned serial collection of respiratory and other specimens and detailing the immune response. We really want to go after what proportion of cases where there is clear exposure might there be that are asymptomatic. So this will be a prospective study of household context of lab-confirmed index cases in households of at least 3 individuals, including children. This is, again, a collaboration with Manish at the Vaccine Evaluation Center. We will be asking the cases and all of the family members to complete symptom diaries. We're debating back and forth whether we will actively collect respiratory specimens at the suggested time points that the WHO has proposed. For sure, we want blood at days 1 and at day 28. And we would rely on that to be able to identify asymptomatic infections, which is our main goal. As I say, we have the funding, but we are adjusting real time the protocol to meet what is the most pressing goal. And as I say, it's a kind of a trade-off. These other objectives, if we go after them, it means less funding than to include more households to ramp up our sample size. So that's all I had to say. I guess we'll move on to hear from Manish and Caroline and Michael.
All right.
Hi, it's Manish. Can you hear me now, David?
Yes.
Okay, perfect. So thanks, Rob and David, for pulling all of us together. I think I just want to start by saying, Rich Lester mentioned that we need data-driven response, and I think underlying all of that will be some really strong collaborations to make sure that our data are reliable and robust. My name is Manish Teddarangani. I'm a pediatric infectious disease physician scientist at BC Children's Hospital. I'm director of the Vaccine Evaluation Center there. Next slide, please. There's my disclosures. Next slide. So just in the interest of time. So while, as of today, we don't have any vaccines for COVID-19, my expectation is that over the coming months and 1 to 2 years, we will, I suspect, have several licensed vaccines available. When I looked at the WHO vaccine trials list, or vaccine development list a couple of days ago, they currently are 62 vaccines in various stages of development, including a few that have already started clinical trials. So the interest that we have at the VEC is on vaccine-preventable and potentially vaccine-preventable diseases, and I would include COVID-19. And we really undertake research across the breadth of vaccine research and infectious diseases. So that includes population evaluation, establishing priorities for new vaccines, discovery work to understand pathogenesis of infectious diseases, assisting in vaccine design, and evaluating vaccine in preclinical studies, both in vitro and in animal models. We do various studies around vaccine development, including vaccine safety, looking at efficacy and immunogenicity, and phase 1, 2, and 3 clinical trials. We also have a lot of interest, and my lab's interest really, in looking at protective immunity. And I think one of the strengths of our position is that we're physically based at the Children's and Women's Hospital, so we have easy access to patients from the hospital. But we also have essentially a research clinic area where we can have people from the community come to our clinic and have samples taken, and including studies in the past where we've done lymph node sampling, for example. So we can do invasive procedures in that sort of setting. And then our lab is up on the 5th floor of this building that you see here, which means that any samples that have a time-sensitive nature in between collection and processing can also be dealt with very easily, and our lab has access to a full range of immunologic measures. And then we also have a lot of research interest in implementation, looking at policy, evaluating public programs, phase 4 trials, and understanding vaccine hesitancy. And this is in collaboration with the other investigators at the VEC, so that's Julie Bettinger, Soren Gant, and Ali Harandi. So next slide, please. So specifically in COVID-19, so in a number of these areas, some of these have been mentioned by some of the prior speakers, so I won't go into detail again. So we're currently part of 2 national observational studies of COVID-19 in children. As we've heard and as we know, children are a minority of disease cases, and so I think it's important that we try and collect as much information as possible from every case. And so through a couple of national networks, so PICNIC, which is a network of pediatric infectious disease physicians, and the Canadian Pediatric Society Surveillance Program, we're collecting data on all children that we come across with COVID-19. Shireen already talked a little bit about the treatment trials that are going on, and we were discussing this morning with collaborators across the country to having a version of CATCO in the pediatric population as well, and as part of that collecting various samples that we can look at in terms of the immune responses. The household transmission study, Danuta has already mentioned, so I won't go into that again. In terms of vaccine trials, so we've had discussions with some of the vaccine companies that are working in Canada on this, including a company in BC, and also through the Canadian Immunization Research Network. And there's multiple trials, sort of phase 1, 2, and ultimately phase 3 trials anticipated, and we expect that some may be starting as early as May. And so there's one trial where we've had an initial review by Health Canada, and also a preliminary review by our ethics board of the trials protocol. And through all of this, I reflect on when I took over as director of the VEC in 2016, and David Shifley almost ceremoniously handed me a small USB flash drive, and he said, "This may be the most important thing I give you, and it contains everything we learned from the research we did in the H1N1 pandemic, and keep it somewhere safe." And so it's been there waiting ever since. We're also doing a number of studies trying to understand immunity to SARS-CoV-2. So the immunity study, which Danuta mentioned, there's also an international study being led by Upton Allen in Toronto to try and look at host genetic determinants in terms of immune responses to the COVID-19. And there's various other studies that we have in planning and have submitted funding applications for, really to do much more deeper immunologic profiling of infected patients to try and understand a little bit more what's happening with the immune response. And I think that'll help us understand the disease. The idea is also to collect similar samples from vaccine trial participants to be able to compare vaccine-induced immune responses to responses in infected patients. And we're also, I think it's great to hear about the trials going on targeting ACE2, because that's a evidence-driven treatment trials. And there's a lot of talk about different treatments targeting the virus or looking at host immunomodulation. And I think having more data to really understand the host immune response during disease will help to refine what the real targets for any immunomodulation should be. So next slide, please. So I think in terms of the key gaps that David asked us to look at, so I think really understanding the groups at highest risk of severe disease and who is really driving transmission in the community, and Danuta referred to that a little bit. And I think really looking at a community level of the asymptomatic cases particularly will be really important to understand that. Trying to understand why people are at high risk, as well as some of the sort of comorbid factors that have already been mentioned, but the underlying immunophysiology of this. What does protection against disease look like? As I mentioned, comparing natural infection with vaccination. And when we're thinking about vaccines, are we just trying to prevent severe disease manifestations, or are we actually trying to block transmission? And that'll really determine what we're looking at in terms of immune responses and mucosal immune responses, but also how any vaccine is implemented in a population, assuming we can understand who is driving transmission. And then also understand a little bit about how long protection might last. There's been various models thinking about, future waves of disease, potential for future seasonal outbreaks. So we'll need to understand duration of protection to be able to implement any preventive measures, and thinking of the implementation end of how a vaccine may be implemented. So next slide. So that's all I have for today, but I have people to email me if they have any questions. Thank you.
Thank you, Manish. Beautiful, succinct overview. We'll go straight into Carolyn and Michael's talk about their modeling, which has been helping us guide an awful lot of the healthcare system preparedness recently.
Okay, great. Michael, did you want to start off? I think you were going to take the first few.
Yeah, thanks, Carolyn, and thanks, David. Good afternoon, everyone. I'm Michael Otterstatter. I'm a colleague of David's at the BC Center for Disease Control. And Carolyn and I are going to split these 5 minutes. quick 2.5 minutes to tell you about some conceptual pieces related to the mathematical modeling work that we're doing. We're part of a mathematical modeling team that's been assembled as a collaboration between BC CDC, Simon Fraser University, the University of British Columbia, Ministry of Health here in BC, and we even have some technical whizzes from the Department of Fisheries and Oceans helping us out. So we have a very skilled team, and I have the honor of working with all of them. And our focus is on, right now, rapid response modeling to inform public health action for COVID-19. Now, the word model is being used a lot in news and in social media. It's something that we hear about every day, and it's used quite loosely. What we're talking about here are the mathematical frameworks that we use to synthesize data, and particularly to estimate things like future outcomes. And the key thing is that these are one tool for decision making. So essentially, the modeling component is taking all of this rich data that we have that comes to us through the kind of monitoring components of our work, the surveillance, the case notification, hospitalization, contact tracing, and outcome data, putting those pieces together, and then using that mathematical set of frameworks and a combination of analytic epidemiology, we generate information that then can be passed on for policy and practice and situational briefs, and for public engagement as well. Next slide, please. And what I'm going to just mention a few of the avenues of work that we have going here, and then let Carolyn expand on this a little bit and tell you also about future directions. Some of the things that we focused on since about the beginning of March have been models such as those for importation risk. Early on, of course, this was a critical aspect. We were curious about estimated numbers of new cases arriving in BC, particularly from overseas and from the United States. That's perhaps less of an issue now as border closures have become a reality, but now we think about importations even from other parts of Canada, and we think that this is going to continue to be important, particularly as we move into sort of the downslope of the epidemic curve. The second area of work that we focus on is forecasting, and we do this on a weekly basis, if not more frequently than that. And what we do is we're projecting new cases based on a number of different models, and one of the key models we've been using is a stochastic model of epidemic growth. And what we do with these forecasts is we pass them over to regional planning folks who also use them for generating expectations of hospitalizations and ICU use. And that's important, of course, from a sort of a capacity planning perspective. And the last area that's of course top of mind for us, and as it is for everyone else, is the kind of social distancing component. So we're interested here in the impacts of social distancing on transmission, and we have a dynamic compartmental model for looking at that. And we assess the effectiveness of the social distancing measures that have been put into place, but crucially the continued need for these measures, and how we can move forward under kind of different scenarios of social distancing. And we inform a lot of this using a variety of behavioral data that helps us to understand how the population is behaving with respect to things like physical distancing. So with that, I'll pass it over to Carolyn to tell you a little bit more.
Yeah, so this shows an illustrative set of simulations from our social distancing model. I should say that the black dots are the reported case counts by day, and one of our huge challenges is really that these are, as you can see, they're very noisy, and we don't always have a great understanding of why the number of tests done changes so much by day. So changing testing criteria, as Danuta commented, is very much confounding our ability to see these trends quickly. So, but I will show you these anyway, and that's partly why these big ribbons are so big. So actually we think now we do have the data to estimate the strength of social distancing that is currently in place in BC, and we're on the panel on the far right, which we call strong social distancing, and we should start to see that curve downwards in the reported number of cases as of a few days ago, and I think we are seeing that. The estimates we've done suggest that we may be doing more than we strictly need to be doing, so that's great that we're not sort of critical in the model in terms of continuing a trend. Moderate social distancing in the middle would see a much slower decline, but still a decline, and then what we're considering weak social distancing, where we're not really controlling community transmission. Of course, these cases continue to rise, but at a higher rate. So those are some of the things that we're thinking about and some of the challenges we have. Okay, can you switch? Yeah, next slide, please. When we're thinking about future steps, of course, we want to understand, if we believe our estimate that we can start to relax things a little bit, at least, that we're well under the threshold, then, of course, that brings up the natural question of how exactly might we want to do that. And that really gets us back to some of the key uncertainties that Danuta mentioned. We don't know what's happening with kids. We know that they're not getting severely ill, but we don't know what they're contributing to the force of infection. So answering a question like, is it safe to reopen schools, or how long do we need to wait before we reopen schools, really depends on population level prevalence by age and on age mixing. So of course we have models of age mixing, and we can make different assumptions about those uncertainties, but without an understanding of prevalence, that's quite inhibited. We, of course, will need, as cases continue to decline, we will need good ways to find cases and continue the excellent contact tracing. One of the things we think is the difference between BC and places like Ontario, Quebec, and for sure the UK, is really that we were able to act early, and by we I mean you, the BC CDC, in testing early and contact tracing early. So those have to continue to play a role, especially as cases decline. Those are some of the things that we're thinking about into the near future with the modeling, and thinking about, what does very strong contact tracing, what would it look like in models. The other key thing I want to leave you with just before our couple of minutes ends is really that right now with the testing that we have, and you saw the variability in those data, one of the challenges we will have in thinking about relaxing measures is that it takes about 3 weeks between a change happening and us seeing it in the data that we have. So I know there's a tremendous audience of incredible expertise here, and if you have ways or think of ways where we could monitor how well we're doing faster than what we're seeing now with that 3-week delay, with that very large amount of noise in those data, I think that would be incredibly helpful in terms of thinking about how well are we doing with a measure, trying a measure and seeing if it's a disaster. What we don't want is try to relax something because of the economic reasons to do that and the other health-related impacts that we're having, and then have to wait 3 weeks and see that we've caused a huge problem. So I think that's a big challenge for us. Is there another slide thanking the people? Yeah, so thank you to everyone on this team. You can see the modeling team there listed, and the surveillance and global epidemiology teams. Thank you.
Well, thanks very much, Carolyn. Really thanks to Danuta, Manish, Carolyn, and Michael. The surveillance and modeling work has been really important in terms of guiding what we're doing, and the knowledge that we're ready to take a look at vaccine candidates as they become available is reassuring. And Carolyn, we've got your message that we need to be able to better inform prevalence in real time, and that's a challenge for everybody in the lab too. So we're moving from epidemiology and public health over to social dynamics, communications, and trust. And we'll be hearing from Cynthia Jardine from the University of the Fraser Valley, then Yue Chan from UBC, and Kelly Lee and Julia Smith from SFU, and lastly, Emily Rempel from the CDC. So Cynthia, over to you.
Hi, thank you very much. My name is Cindy Jardine. I'm a professor in Tier 1 CRC and Health and Community at the University of the Fraser Valley. I'd really like to acknowledge my co-investigators on this study, Dr. Martha Dow and Dr. Satwinder Baines from the University of the Fraser Valley, Dr. Michelle Dreger from the University of Manitoba, Dr. Anita Haywood from the University of New South Wales in Australia, Leslie Gray from the University of Otago in New Zealand, and we're really happy that we have both Dr. Bonnie Henry, the Provincial Health Officer, and Dr. Alex Choi, who's the Medical Officer of Health for Fraser Health, as knowledge users on our team, and that we have the support of the Divisions of Family Practice in the Fraser Valley. Next slide, please. So this is something completely different than what we've heard of so far. We're looking at travelers, and we're specifically looking at a class of travelers that are known in the literature as VFR travelers, with VFR standing for Visiting Friends and Relatives. We have a large number of Canadians who have chosen to settle here from other countries of origin and understandably travel back to those countries of origin on a regular basis to visit friends and relatives. They are particularly susceptible to infectious diseases of all kinds, not just COVID-19, because they travel differently than other people. They go to these countries, they live as locals, they often think they're not at risk because for many of them they're going home, a place that doesn't feel risky. So they are well acknowledged to often be at higher risk of contracting infectious diseases. They also account for more than 2 million visits outside of the country every year. So what we're trying to do in our study is to get a better understanding of their awareness, their knowledge of infectious diseases and COVID-19 in particular, and the measures that they take to protect themselves when they travel. We're looking at their information-seeking behaviors, including whether they seek pre-travel advice. And then we also want to know whether family physicians who may be providing this pre-travel device, the types of knowledge they have, the kinds of experience that they have in dealing with these kinds of travelers, and their needs to be able to do their job better. Next slide, please. So we're looking at 3 populations, primarily in the Fraser Valley. We are looking at migrant populations. We're probably going to be specifically focusing on both the Chinese diaspora and the Punjabi diaspora, simply because they make up the largest population of migrants in the Fraser Valley. We're going to complement this by looking at international students. And my university, the University of the Fraser Valley, has a large proportion of international students. And we're also going to talk to family physicians in the Fraser Valley and the Lower Mainland. Next slide, please. In terms of our outcomes, we'd really like to understand the travel practices of this group of people better, whether they seek pre-travel health care and advice. We do know that the province of BC, as well as most provinces in Canada, no longer cover that under health insurance plans. So it makes it sometimes more difficult for them to get the kind of advice that they need unless they're looking elsewhere. And how difficult it is for them to access resources to protect against disease transmission. We'd like to turn all of this into actionable guidance, so we have better risk communication strategies and better community engagement to make sure that our regular travels are more ready to travel responsibly and safely. And finally, we're lucky enough to have 2 international members of our team. We are actually modeling a lot of our work on that, done by Dr. Anita Haywood in Australia, who's a world expert in the area of VFR travelers, and that'll help us situate our results globally. If I could add a couple of other things, of course, we devised all this in the 2nd week of February when travel was one of the major considerations of COVID-19. It's obviously not the most pressing problem right now, but we're forging ahead. We had also planned to do a lot of this through focus groups and interviews with people. And all universities right now are under restrictions for in-person engagement of research participants. So we're having some interesting discussions on how to do this, especially in doing focus groups that require translation and how that looks. We have launched our survey of students at UFE, and we've expanded this actually to include all students at UFE and talk to them about their international travel knowledge, practices, behaviors, and their specific knowledge of COVID-19, and what they think about COVID-19 communications that are coming from their institutions, taking advantage of things that have happened since our research was funded. So thank you very much.
Thank you, Cindy, and your UFE team. Great to have you on board. Yue.
Yes. Hi, everyone. My name is Yue Chen. I'm an assistant professor of sociology at the University of British Columbia. Sorry, like the title of my presentation is wrong. I'm going to speak about the social impact of COVID-19. Definitely changing marital status and family relations is a dimension of social impact. Next slide, please. So my project has been funded by CIHR. I want to acknowledge my collaborators. Amy Hanser, she is Associate Professor of Sociology at UBC, and Wen Fan, she is Assistant Professor of Sociology at Boston College in the States. And I have two excellent research assistants, Suki Xiao and Rose Zhang. They are both graduate students of sociology at UBC. Next, please. So my research mainly focused on Wuhan, the former epicenter, where the first case of COVID-19 was identified, partly because I was born and raised in Wuhan. So I knew about COVID-19 very early on and have been following the development. And I do think because we have a 1.5 to 2 month lag between kind of the COVID-19 trend in Wuhan, then in North America, what we learn from Wuhan can inform our decision making in North America. Next. My research is mainly focused on human experiences and mental health consequences of quarantine. And in North America, we call it social distancing. And so I have written an easy to read article published in Context magazine. This is an outlet for sociology for the public. So the article is about social isolation and city shutdown as a response to COVID-19. In this article, I propose that we need to pay attention to the cultural dimension of outbreak response because a large-scale quarantine was easily implemented in Wuhan, but social distancing measures have been more difficult to implement in Canada and United States and Europe. And in addition, learning from the experiences in Wuhan, community and neighborhoods play a very important role in enhancing the residents' capacity to cope with the quarantine. So we need to think about how we can engage communities to form coordinated, localized help for our citizens. Next, please. And my research focus on particularly 6 groups. The first 3 groups: pregnant women, elderly living alone, people with chronic conditions. They are people with special health needs, healthcare needs. And then the other 3 groups: parents of young children, healthcare frontline healthcare workers, and family members of COVID-19 patients. They are primary caregivers. So we really want to think about how the intersection of work and caregiving can influence individuals and families' experiences during and after the outbreak. Next, please. In terms of looking ahead, right now I'm collecting data, so it's a very interesting time because we also need to kind of collect the data while there is a travel ban. I really hope that more sociological and social science research can be done to aid post-pandemic recovery and also think about the social impact of COVID-19, because COVID-19 really challenges a lot of assumptions in our infrastructure. Right now, everyone is working from home. The assumption is that everyone can work from home, and it doesn't matter if kids, and we have kids at home, and everyone can work. But really, those COVID-19 give us an opportunity to reflect on a lot of the assumptions in our policy and moving ahead, how we should develop or modify our policies to respond to future other pandemics or outbreaks. That's all. Thank you.
Thank you, Yue. We'll go straight to Kelly and Julia.
Great. Hi everyone. My name is Kelly Lee. I'm a Tier 1 CRC in Global Health Governance in the Faculty of Health Sciences at SFU. Also a UBC graduate in International Relations, where I'm leading an international team to compliance with the international health regulations. Our team is based at SFU, University of Maryland, Sydney, and Hong Kong. And we've been funded through the first tranche of the CIHR Rapid Response funding, the New Frontiers in Research Fund. And our team includes knowledge users as well, which are important, and their logos are there. And our interest primarily is improving coordination of the outbreak response globally. And by globally, I mean across the world, including Canada. Next slide, please. So our research begins with recognition of the abundant evidence that clearly shows that coordinated action within and across countries, as well as across different sectors, that is health, transport, education, and so on, and also across public and private sectors, is critically important to achieving timely, appropriate, and effective outbreak response. And in contrast, poor coordination based on individual action within countries leads to less effective action. It's probably quite intuitive to think that. So our research is largely focused at the global level, but it's important to recognize that global and local action is very much intertwined, and we decided to, what we decide to do in BC, as we've been hearing, matters nationally and globally, and vice versa. What we do globally also has consequences locally. Okay, next slide, please. So the basis of international coordination, the global level, is the international health regulations. And as a reminder, the IHR is a legally binding instrument of international law under the auspices of WHO. And it sets out the obligations of member states of WHO when dealing with the international spread of diseases and other health risks. And these obligations are based on specific principles, which you see on the slide. And the challenge, WHO does not have the formal mechanism by which to enforce the IHR. And so what we have is an inconsistency of practice and in some cases non-compliance. So what we're seeing already is that, for example, WHO recommendations are widely ignored so far by many countries. 75% of WHO member states have actually adopted travel and trade restrictions compared to previous public health emergencies, such as the Ebola outbreak, which had about 25% of countries. There's been quite a lot of variation in what and when cross-border measures are adopted, and then there's high variation in what and when beyond the border measures have been adopted within countries. Okay, next slide. So our research is addressing a number of gaps. We are seeking, first of all, to understand the full range of cross-border measures that are being adopted. We want to review the evidence behind these measures, if it is available, on the public health impacts, but also the wider impacts on society, and that includes economic impacts, but other impacts as well. We're going to conduct 4 case studies to understand better how these decisions are made. When you adopt a cross-border measure, what's going through the minds of decision makers. And then finally, ultimately, we want to create a decision tool that encourages increased compliance with the IHR, and really it's about better coordination globally. So my last slide just shows what we hope to create in terms of resources and tools. One is a data set on cross-borders, and we're already started collecting that. We actually have an existing data set which is looking at previous outbreaks, and we're adding to that, so that's why we can compare across public health emergencies. We want to create some or develop some rapid and systematic reviews of the evidence, as I described. We've already done one for WHO on travel restrictions, so we've provided that already to WHO. We're going to conduct these 4 case studies: Canada, Hong Kong, Australia, and the US, and what's happened over time in terms of decision making. And finally, this is probably where we need some help, and we would very much welcome partners. We're looking at applying to the digital technology supercluster, try and find partners to create a data analysis software platform of some kind to help us provide a tool by which decision makers can make better decisions in real time about what kind of cross-border measures to adopt, when to adopt them, and also when to get rid of them, which is also equally important. So that's what we're trying to do with our project. Thank you very much.
Terrific, Kelly. And Julia's ready to go?
I hope so.
Hi, everyone. I'm going to share about a different project. I also work with Kelly, but I am leading a project on gender and COVID-19. And this is a global project as well. And we are looking at different gender impacts of the epidemic and of the response here in Canada, in the UK, in China, and in Hong Kong, as well as more globally, and providing policy analysis and working with policymakers to inform responses to mitigate those impacts. So we set out our ideas around what we can understand about, or we can predict would be the impacts related to on different genders, based on research that different people in my team, and you can see the team there, have done on past outbreaks like Ebola and Zika, H1N1. And lots of these relate to the fact that women make up the majority of the healthcare workforce, and so are on the front lines of the outbreak, that women provide the majority of care work in the home. Of course, with this particular outbreak, we're also seeing higher rates of mortality amongst people who identify as men, and so that's something that also has to be considered. We're also seeing already increased rates of gender-based violence and violence in the homes, where there's social distancing. So, for example, today there was a media report that the Women's Health Center helpline in Vancouver has received a 300% increase in calls about domestic violence. So what we're trying to do is gather data on this, and this is partly because this has never been done in epidemics. Those of us who have researched past outbreaks and gender impacts have always done it after the fact, and so here we have an opportunity to try and collect data on what's happening now. Next slide, please. So one of our case studies is Canada, and we will be focusing on analyzing gender impacts here in British Columbia specifically. We're using a gender analysis framework, and I've put an empty framework up there yet because I don't have enough data to fill it up yet. But what we'll be doing is looking at, okay, what data is available about vulnerability to infection, illness, and these different gendered impacts. What data is available? What examples do we see? What can we know about differential impacts to access to resources during this time, about changes in labor, norms, power, and security? So we're all social scientists, and we're approaching this from international relations, political economy, sociology, health systems, a whole range of perspectives. The team's interdisciplinary. But we do hope to be able to produce this sort of gender matrix, which we'll be sharing online and updating at least monthly, documenting the real-time impacts of the outbreak and of the response on men and women as well as other groups. Tied to this will be a toolkit that will provide indicators, will provide suggested responses and recommendations for those responding to the outbreak, as well as provide analysis of where the gaps are. Next slide. Time. Thank you. So gaps in research, there's so many, I could go on for a long time. The one I will focus on, because since we have the BC CDC here, is the desperate need for sex disaggregated data on cases, hospitalizations, deaths. I should have had testing in there too. We're working with the WHO right now on global data sets, and they only have about 10% of data on 10% of cases. We really need to understand these impacts more, and you can see the other gaps there. Next slide. In terms of resources, we have established a gender and COVID working group, and there's an email there for the person who's coordinating that. In that working group, there's about 55 people from around the world, ranging from biomedical scientists to medical historians. It's really an interesting group if you're interested in this. We also have compiled a gender and COVID resource document, and we'll be developing more stuff soon. Thank you.
Thank you very much. Yeah, that's good. We will run straight into Emily.
Good afternoon, everybody. I am Dr. Emily Rempel. I'm a knowledge translation lead at the BC Centre for Disease Control. My co-investigators on this project at the CDC are Katie Fenn, Director of Quality, Safety, and Accreditation, and Harlan Pruden, Educator for the Chima-Mook program and also a doctoral student. Our project is on understanding knowledge, attitudes, and behaviors around misinformation and personal protection for COVID-19. Next slide. So let's get into it. Why are we interested in misinformation? So we, as a team, see misinformation alongside active public engagement and consultation as a key gap in current COVID-19 research. Some of us will experience COVID-19 either through becoming ill or, for us on this call, probably more like through our work, but that is a very small percentage of the BC population. Every single other person who doesn't become ill experiences COVID-19 through information and media. So alongside our response to the pandemic, which we've seen great examples here today, we are rapidly responding to an epidemic of misinformation, or as the WHO declared in February, an infodemic. So misinformation can lead to health-harming behaviors as well as stigma and racism. Some examples of that are taking medications because someone says it might help. For example, we've all heard of the controversy around hydroxychloroquine, and even things like making your own hand sanitizer, which can put people at a greater risk of infection because it's challenging to make successfully at home. So misinformation is not just spreading memes on Facebook, it's encouraging behaviors that hurt our mental and physical health. So working out of the BC CDC, we are conducting a rapid response project to proactively address this infodemic as it relates to COVID-19. We have two main objectives. The first is to understand the knowledge, attitudes, and behaviors driven by COVID-19 misinformation, and second, to understand where British Columbians access information, what sources they trust, and how they prefer to receive messages about COVID-19. And we're going to do that through a cross-sectional rapid response online survey of 3,000 British Columbians, reflecting diversity and inclusion. And what I mean by diversity and inclusion is that we're actively ensuring our survey is a safe and open space for people in LGBTQIA2S+ communities, in particular using language that is welcoming and open. So summing everything up, we want to know how to get the right information to the right audiences in the right way at the right time. Next slide, please. So what do we mean by a rapid response? We are conducting this survey with the market research firm Lux Insights, and it's funded by the Michael Smith Foundation. The firm is rapidly turning around both the survey design, which we're working with them on, and the initial analysis for us in order to inform our communication and engagement strategy in near real time. So this is essentially a quality improvement project that will hopefully lead directly into public health intervention very quickly. And we have 4 key theoretical frameworks that we are working with. First and second are related, and they are knowledge exchange and public engagement. Essentially, if you want to communicate with the public, you should ask them what they want to know about. Third is Indigenous research. This means collaborating with Indigenous communities and researchers, and Harlan Pruden and Jessica Chenery from Chima-Mook at the BC CDC are leading this aspect of our research. And finally, feminist research. And this means responding and researching at the same time. Feminist research means both knowing and doing. We want to break down the idea that we are the experts at knowing how to communicate and translate information. Instead, we want to reformat that to thinking, without information, publics cannot make informed and healthy choices, but without knowing what information publics want, we cannot make informed and healthy choices as well. Next slide. So very briefly, our research is looking at 4 key dimensions. First is communication preferences, then COVID-19 knowledge and beliefs, then both harmful and helpful coping strategies and behaviors, and finally stigma and racism. And overlaying all of these dimensions is the concept of trust. Trust is often incorrectly thought of as a yes or no phenomenon. Either I trust that person or I don't. But trust is actually critical, and it's strongly related to communication. And people are going to vary their trust in us to the degree that we demonstrate that we are trustworthy. So this survey provides not just a tool to show that we are trustworthy, we want to talk to people, but also to show that we're considering trust in how we communicate. All right, so next slide. So what resources do we have that can be shared from this? First, our data can be used as secondary data within our response efforts. but we are also a resource for people who are interested in asking complex social questions with Indigenous, public health, and government partners at the BC CDC. And I've listed all of my collaborators here for that reason, and they are all amazing. Thank you very much, those who are listening in on the call. But to end our story, as I mentioned, most people experience COVID-19 through information, and we want to keep it that way. If they only know about COVID-19 through media, that means that they are not ill. They have heard our messages, and they are doing things that limit their risk of infection. So to maintain that phenomenon, we need to address the infodemic as part of the pandemic. Thank you.
Thank you very much, Emily. Nice session. We'll turn it over to Rob to introduce the final session on therapeutics and vaccines.
Good. Well, David just introduced it. It is on therapeutics. We have a number of talks on therapeutics and a couple on vaccine development and some on infrastructure. So we actually are a little bit over time, but we actually have this Zoom account actually open till 5 o'clock. So we do have time, and it is being recorded if you have other events to go to. From some of the talks, a couple of them are talking in pairs. I think we have 8 talks. One of them is quite short. So there'll be, now we're introducing some really innovative therapeutics and innovative approaches for vaccine. And the first speaker is Bo Barnhart from AbCellera, which I think he'll introduce AbCellera himself.
Yeah, I will. Can you hear me okay?
Very well.
All right, great. Thank you very much for the opportunity to talk to you today about AbCellera's Rapid Pandemic Response Platform. My name is Bo Barnhart. I am a scientific director at AbCellera. And what I'm going to do today is start off by talking to you a bit about our platform and technology and explain why we're well suited to tackle a pandemic of this type. I'll show you some examples of that, a test that we did just last year of the platform in a demonstration of the ability to potentially take on a pandemic, and then ultimately what we're doing now for the COVID-19 pandemic. So next slide, please. AbCellera is a biotechnology company here in Vancouver, and it is based on a high-throughput single-cell microfluidics screening system that was developed by our CEO, Carl Hansen, while he was at the University of British Columbia. And what it allows us to do is very deeply and very rapidly screen natural immune responses from essentially any organism. So we do that in our discovery space by immunizing model animals. But of course, for a pandemic response, we're doing that with the natural immune response from humans, and particularly humans who have recovered from the disease. And so what we do is seek the antibodies that provided some protection to those humans and allowed them to overcome the disease by applying this platform and this technology. We have a world-class and really unprecedented depth to which we can seek those antibodies from single B cells. When we complete a screen, we very rapidly take those single cells and move into our single cell sequencing. We profile them, pull the sequence out of that B cell that's producing an antibody that performs to the specifications that we are interested in. And then that allows us to take that very large data set of antibodies and the sequences of those antibodies to do much more in-depth analysis downstream, kind of if you kind of follow down this pipeline using our proprietary visualization bioinformatics software system called Symposium, which allows us to go through that very large data set and understand what kind of responses we're getting. So speed and depth are two of the really important pieces here. We can go through 1 million cells or more per day and very rapidly turn that around into sequence results, looking at hundreds of sequences within a couple of days. Next slide, please. Demonstration of this response was in fact completed just last year. We used the target flu for this capability demonstration, and this was in collaboration with our partners DARPA, the U.S. agency, as part of their pandemic prevention platform. And you can see at the top there the goal, find the virus, find antibodies to that, and then ultimately generate a therapeutic. So I'll show you a couple of examples. If you go to the next slide, please. For the sake of time, I don't have sufficient time to go through the entire process that we did during this capability demonstration, so I'm really going to focus at the beginning on screening because I think there's a good application to what we're working on now with the COVID-19. But the goal was to start on day one with a sample, in this case a mock sample from a mock patient from a pandemic. And we set out to go through each of these steps, ultimately having identified antibodies that protected mice in an animal model from lethal challenge, from influenza, all within 65 days. And so to start at the beginning, which is the screen, you can see there a schematic of how we do the screening. It's cell-based, so cells expressing the protein on the surface that is expressed by influenza, in this case, hemagglutinin. And a single B cell in each chamber produces antibodies, and we're able to detect the antibodies that bind to that hemagglutinin on those cells specifically. On the bottom are actual images from that. And you can see the second to last one is the one where we're detecting those anti-IgGs. If you go to the next slide, just a quick demonstration and description of the success of this capability demonstration. So we had success at each and every step. If you look at the end first, we accomplished this in 55 days. We looked at almost 1 million cells, more than 4,000 hits, almost 500 unique sequences representing more than 200 clonal families. And then in the progression through the analysis and characterization of these antibodies, identified 7, took them into mouse models, and all 7 of them were completely protective against a 20x LB50 animal model, so success at every step. Those stars above are, I think, what we're experiencing now as I think, particular gaps within COVID-19 research. The proteins expressed by the SARS-CoV-2, important to make sure that we have the appropriate ones and are accounting for mutations, as we've heard a little bit earlier. In vitro neutralization assays, so really looking at viral blockade, has become an important place that I think we need some additional opportunities, as well as animal models. If you go to the last slide before the acknowledgements, I'd like to just give you a quick rundown of where we are with COVID-19 in the last minute. We started a screening campaign, so you can imagine what we learned and all the expertise and things we picked up over the capability demonstration were very nicely applied here to COVID-19. Immediately realizing that there was an opportunity here, we received a sample from one of the first patients in the U.S. to have recovered from COVID-19. We ran through the same kind of set of steps that I described earlier, particularly at the beginning. Discovered and sequenced more than 500 antibodies, very quickly moved into functional testing with our collaborators at the NIH. We also teamed with Eli Lilly, a large pharmaceutical company, to develop, manufacture, and distribute potential therapeutics. And in the timeline, it took us 3 days to go through about 6 million immune cells. We identified over 2,000 antibodies. 2 days we did some sequencing, quite a bit of it, and I identified 500 unique human antibodies. And in total, 23 days after receiving the sample, we had 24 lead antibodies with behaviors that we were interested in continuing to progress. So I'll go on the last slide. I'll stop there, please. So I want to thank everyone who's been involved in this. And reach out if there is interest in potential collaborations or places where we may be able to help one another. Please reach out. There's some contact information there.
Good. Thank you, Bo. I think you've heard already a number of epidemiology studies, addressing immunity in long-term, which I know you've already made contacts. It'd be a rich resource across BC. This is what we really are encouraging, is this type of team science and collaboration. The next is now a different type of drug development, is Art Cherkasov from UBC and the Vancouver Prostate Center. Art?
Yeah, hi there. I'm Art Cherkasov, so I'm with UBC in urology, and we're working on precision drug discovery in cancer. But before that, for 7 years, I was with infectious disease, and now I'm somewhat turning back into my early days. And so we are looking into developing small molecule therapies against COVID. And we are related translational, so we have a very established track record in developing new drugs. But today it's not about my team, it's about our bigger team now, where it's me, and I'm lead in silico drug discovery components. And there is Natalie Struniatska, who is leading our structural biology studies, because structure of target proteins is very essential. And Dr. Francois Jean, who is providing all virology screens and support. So 3 of us joined forces more than a month ago. We applied for this CRHR opportunity. and we've been successful. And the project is really augmented discovery of new inhibitors for COVID. And by augmented, I mean we've developed a new protocol which enabled us to go through almost 1.5 billion small molecule candidates for our main protease target in COVID in less than 2 weeks. That's how we really, and I did it in February, that's how we really got ahead of the game, because we are probably anywhere 5,000 to 3,000 times faster than any competition. And so next slide, please. We already identified a short list of compounds which might inhibit main protease in COVID, and compounds already been ordered from our side. Natalie is preparing crystals of the target for high throughput screening, and Francois is setting all the assays on campus, on level 3 facilities. So we are trying to build a pretty complete pipeline, which is synergetically unifying all our expertise and skills. And it's being scaled up by every week pretty much. So we get more compounds, we get more targets as well. So just to mention now, in the last month, we worked 24/7 in silica group. Now we enhanced our capability tenfold. So now we are ready to screen 13 billion molecules using computers against any particular target. And so our timeline is to generate few hundred candidate compounds per target within several months and screen them as well. So next slide. So really, the only gap is only need for funding, because this small molecule drug discovery business, that's where dollars buy time. So as I say, things really get scaled up, and I can say that the world is turning to us as well. So a lot of people want to work with us because we have expertise, and as I said, we put together the complete pipeline. So we get a lot of different ways and proposals to screen more chemicals, and also number of target multiplies. So every week there is at least one new target being resolved with good resolution in COVID, so new protein to look at. If we started with just main protease a month ago, now we have 5 pretty credible targets. So basically in a month, our small molecule value volume went from 1.3 billion to 13 billion, and number of targets went from 1 to 5. So we have to scale up. That's why we need funding, and we're looking actively for all kind of opportunities. But that's really the only gap in our operations. Everything else is pretty much well under control and being put together. And next slide, in terms of resources, we are building the entire pipeline, which starts with all kind of small molecule feed-in. That could be molecular databases I mentioned, which again, tens of billions of molecules. With some collaborators, we're also establishing some text mining capabilities, so we can, in fact, we'll be extracting absolutely every small molecule mentioned in literature in the last few months in regard to COVID and associated with the target. And so we have a pretty sophisticated international triaging system now, and our in silica and experimental capabilities could be part of that triage to really go through everything to a few molecules which will have a chance to actually go and pass clinical trials. Again, small molecule will take time and resources, but we definitely have everything together. It's a very strong team, and we are, again, working tirelessly on this day and night. That's all. I'm at the end of my 5 minutes now.
Thanks, Art. I think we have a team next, Eric, Dan, and Francois, John.
It's just actually it's just going to be me talking about a team grant that we got with Chris overall. So it's Francois at a different time. So thanks for this opportunity. So I'm a professor in the Department of Biochemistry and Molecular Biology, and this is a team grant that we with Chris overall that we just recently got from CIHR COVID-19 grant and the rapid call. So Chris overall is a professor in the Center for Blood Research. He is a CRC Tier 1. And we're just combining our expertise. My expertise is in molecular virology, and Chris is in degradomics proteases and developing novel proteomic approaches. So the objective of this grant is to identify the host cellular proteins and substrate specificities that are targeted by SARS-CoV-2 proteases. And the end goal is to generate antiviral consensus peptide substrate analogs that block viral protease function and infection, but also to gain a more complete picture of how COVID-2 infects cells, and that can lead to COVID-19 disease. So next slide, please. So this video slide, you can concentrate on the left-hand side. So Art described the drug targets, why we go after the 3C protease. I just wanted to remind you that these proteases, and there's two of them in COVID-2. There's a PLP-Pro, which is papain-like protease, shown in blue, and this 3C-like protease, shown in red here. And so these viruses produce a long protein, and essentially they need these proteases to act like scissors to cut the long protein to individual mature proteins, shown by those arrows in the blue and the red. So we have a very good understanding of that aspect of it, but there's a big gap in knowledge here, and it's very well known in other viruses, viral proteases target cellular host proteins in order to, in a strategic manner, to modulate host pathways and disable antiviral factors to promote virus infection. And so what we don't know here is that these, we don't know any of the cellular targets, for the most part, for these proteases. And so what we are using is a proteomics approach invented by Chris Overall called TAILS. It's terminal amine isotopic labeling of substrates. And on the far right, top right there, it's just a depiction of, say, a cell with a bunch of different proteins, and just showing you a scissor basically representing one of those proteases that can target one of these proteins in a cell. And essentially what this proteomics approach is, a very clever one, is sort of like finding the needle in the haystack and trying to get rid of all those other proteins, and then just targeting and identifying just those proteins that are targeted by that protease. So we can identify the target proteins and find the amino acid sequence that those proteases recognize. We're taking a two-pronged approach, is using purified proteases as well as to look in COVID-2 infected lung epithelial cells for now. And we want to identify, this will identify the direct targets and also capture the proteolytic signature in COVID-2 infected cells, which is very important for uncovering key viral host interactions. Next slide, please. And so the major outcomes is, one, to determine the consensus substrate specificities of those proteases. Basically, we would identify the sequences, amino acid sequences that are recognized by these proteases, and then we can turn that around and then generate inhibitory peptide analogs that can block protease function and hopefully infection. And we conjugate those peptides with fluoromethyl ketone, which will block that catalytic residue. We also will have a more complete picture of how COVID-2 leads to infection and COVID-19 disease. And what we believe is that those proteins that are cleaved by the protease, the host proteins that are cleaved, are strategic and are important for promoting virus infection. So this is important knowledge that will reveal novel host targets for antivirals that can lead to host-directed therapies, and then ultimately lead to more preparedness for future outbreaks of coronavirus infection. So we have a number of resources that can be shared. We have recombinant proteases, induced small expression plasmids. We are working on a number of different coronavirus infection models. These murine coronavirus models have been excellent. Some of them mimic some of the symptoms that we see that are in SARS patients. We have the human coronavirus that can lead to a common cold. And obviously, we have COVID-2, which is being brought in by Francois Jean into the CL3 facility. We also have GFP reporters that can sense the protease activity, so they fluoresce upon protease activation. And this is quite important. It's been generously provided by Nevin Krogan and Volker Dial, where basically we have all the open reading frames for COVID-2 in expression constructs, as well as the infectious clone, which we now can do reverse genetic approaches and test different mutant viruses. That's all I have, and thanks for giving this opportunity.
Thank you, Eric. We'll hear a little bit about the level 3 unit at the end of this session. So we're moving on now to Horatio Bach from UBC in Vancouver Coastal Health. Is that right, or is Francois separate?
Yeah, it's me, Francois. Yep.
Oh, sorry, Francois.
No problem at all. I think...
now we'll hear about the in vitro work then. Thank you.
Okay. So thank you for the invitation. So I'm Francois. I'm a molecular virologist at UBC in the Department of Microbiology and Immunology. I'm the past scientific director of UBC Finder. The current scientific director is Prof. Eltis. I think that's going to tell us about Finder in a few minutes. My lab is basically studies emerging human enveloped pathogenic viruses, so risk group 2 and risk group 3 agents. We develop host and virus-directed antivirals. For the last 2 years, we have been developing human organoids for our antiviral drug discovery projects. This is done in collaboration with stem cell technology at UBC, and we also work on advanced diagnostics for viral diseases, including a quantitative proteomics assay in collaboration with Prof. Foster here at UBC. So what I would like to do, and my teams are basically funded with 3 operating grants, 2 from CHR, and this week we just heard about our grants from Genome BC basically being funded. So there's the next slides, please. Yeah, one more. Okay, so there's we use 3 different strategies for the discovery of SARS coronaviruses drugs, which are described there. You can actually, this is coming up from a recent Science paper, which I really like, underlining some of the key targets that are under investigation. Those targets include not only direct acting antivirals, which go against the virally encoded proteases or enzyme, but also there's some of those targets that are host targets. So those then would be fit into the category of indirect acting antivirals. So the 3 different strategies that the 3 different teams that I'm part of are basically using, the first one is the so-called drug repurposing, which is basically conceptually the rescreening of previously approved FDA drugs against other diseases, against basically the SARS-CoV-2 coronaviruses. The main advantage, of course, of this approach is that if you do identify good lean molecules, this molecule obviously is already approved. All of the pharmacokinetics and the safety is basically resolved, and then you could proceed much more rapidly towards basically further clinical studies. The second strategy is what we have been doing in my lab for about 15 years now is host-directed antivirals. In this specific case, we focus on a very important cellular enzyme that is known as TMP-RSS2, which is critical for the activation of the glycoproteins of the SARS-CoV-2. So the cleavage event by this cellular protease is required to expose the fusion domain, which is important for the fusion process and the release of the infectious viral RNA. Our collaborator on that proposal is actually just published in Cell 2 weeks ago the validation of TMP-RSS2 as a target for SARS-CoV-2. So more on this in just a second. And the third strategy is based on the idea that is underlined by both Eric and Hart on targeting viral enzymes such as the viral protease, but there are also other groups across Canada that target the polymerase and the helicase. So the main limitation, I will say, from this approach is the potential of basically drug resistance that are associated to targeting of viral enzyme in general. So the main objective for us then from the output or the results of those studies is to really start to think about combination therapy. So combination therapy that would be involving not only host-directed antivirals, but also will be involving viral-directed antivirals, and hopefully, if successful, other drugs that may be targeting different other steps that we may found from the drug repurposing strategy. In the next slide, please.
what I really want to point out is the investigators that are involved in those specific projects that may help you in terms of potential collaboration. In the project from Genome BC, this is involving as co-investigator Dr. Raymond Anderson and Leona Foster, both contributing from the drug discovery aspect and the proteomics quantitative assays that we're going to be using. I want to underline two important international collaborators, Dr. Barry O'Keefe providing additional FDA-approved drugs or drugs that are currently being tested in clinics for our drug screening. An important contributor and collaborator in this project is Stefan Pohlmann, who, as I mentioned, just published 2 weeks ago a paper in Cell reporting what we call pseudotyped viruses expressing the SARS coronaviruses. And I think the important point to raise about those pseudotyped viruses is that they can be used in CL2 environment, and they are very powerful tools for doing screening for entry and maturation inhibitors. So this is really, really a great asset. And I have to again, also underline that Prof. Pullman is really eager to collaborate and share his molecular tools. So I really encourage you to contact him. Even if he's very much busy right now, I think he's always open to help out. And the testing of those molecules are basically done in organoid-based technology in collaboration with stem cells. So the next slide, please. Time's up. Yes. Well, I just want to underline here that for this proposal from CHR, another important collaborator to keep in mind if we want to move and proceed with small animal model of SARS-CoV diseases is Dr. Gary Whitaker at Cornell University, who is really leading the way right now in the development of such a model, and is also very much open to collaboration. And the last grant is, the next slide is the one that just was described by Hart, so I don't need to add more information on that one. And of course, all of this is will be done at UBC Finder, and the SARS-CoV-2 viruses are actually coming from the University of Toronto and the National Microbiology. So the hard part, I think, is done now at the NTAs, and then the last part will be the licensing from UBC to receive the virus from Winnipeg or Toronto. Thank you.
Thank you, Francois. So now we'll move on to Horatio.
Hello. Yes. Thank you for the invitation to share our proposal. My name is Horacio Bach, and I'm affiliated with the Division of Infectious Diseases, Faculty of Medicine. And my lab is located in Immunity and Infection Research Center, IRC, in the area of Jack Bell Research Center in VGH area. I want to add also that this grant that we are funded with by the CIHR is also Dr. Ted Steiner, co-PI, and he's the head of infectious diseases in Vancouver. Next slide, please. So our project basically is based on the development of a therapy where we will use engineered antibodies to neutralize the virus. Next, please. So our working plan is to screen antibodies, and the antibodies we develop here are called single-chain antibodies. Just a few words, single-chain antibodies are only an amplification and shuffling of all the regions that are present in the variable region of the heavy and light chain of the antibody. So we don't have any constant region. It's only the variable region that makes the antibody very small. It's a 27 kilodalton protein. And as I said, they're engineered by PCR, and we can sequence also the gene. These antibodies are screened and produced using biotanning, and we use E. coli as the target. We don't use any animal, and we can do that in a very fast way. probably in a period of 3 to 4 weeks, we can have the candidates. Definitely, the second part will be once we produce this antibody that we use that consider like a recombinant protein because we can express in E. coli or insect cells or egg cells, and then we can check the affinity of the antibodies to the peptides in this case. Our targets are basically a panel of peptides that they were provided by bioinformatic groups, and they are exposed to region of the S1 protein, basically. The next step will be to test these once we know the affinity of these antibodies for the virus, we will test them in a cell line, using a cell line infection that we propose to use the level 3 or finder in the campus. And definitely the last step will be the animal model to show the efficacy of these antibodies to neutralize the virus. The idea basically is to block the entrance of the virus to the host cells. Next, please. So what we have is an antibody engineering facility at IRC. We will have access to the CBL3 for cell and animal model test, and we have in our lab the capability to produce antibodies at small and medium scale in the lab. And the next, please. That's it. Very simple.
Thank you, Horatio.
You're welcome. Thank you.
So our next is Serum Subramanian, our new CERT chair at UBC. And we'll be discussing the applications of cryo-EM to the study of COVID therapeutics. Thank you, Serum.
Good afternoon, and thank you, Rob, for including me in the symposium. Cryo-electron microscopy, or cryo-EM for short, is an emerging technique for imaging proteins at atomic resolution. Using these methods, 2 groups in the US have recently reported atomic structures for the SARS-CoV-2 spike protein. I am a newcomer to COVID-19 research. In my presentation, I will focus mainly on gaps in imaging that we need to fill in order to obtain a better understanding of the biology of COVID-19, which in turn will help with the development of effective therapeutics and vaccines. The next slide, please. The 3 topics I will touch on briefly are, first, an introduction to the kind of information that we can get from imaging enveloped viruses such as SARS-CoV-2. Second, how this type of imaging can be helpful practically. And third, the type of imaging studies that will likely be necessary to understand how viruses are transmitted following initial infection. Next slide, please. On the left is an electron microscopic image of HIV, which has spikes that stick out on the surface, much like SARS-CoV-2. And on the right is a 3D rendering of the virus determined from cryo-EM studies some years ago. Color-coded to show the spike protein in red, the viral membrane in blue, and the genome in yellow. Next slide, please. In work that we did starting about 10 years ago, we showed that 3D images like this could be computationally processed to obtain molecular models for exactly what was being displayed on the surface of HIV. More importantly, as shown in the next slide, these studies also enable analysis of the kind of molecular movements, next slide please, it showed the enabled analysis of the kind of molecular movements that take place at the instant that HIV contacts the cell surface and are crucial for the viral genome entering infected cells. In this instance, the binding of CD4 opens the spike, enabling the fusion peptide to insert into the target cell membrane. As shown in the next slide, studies like this are useful to determine not just how receptors engage, but also to understand how antibodies that block entry work, because they help us identify exactly where the antibodies bind in the context of a native virus and the specific protein conformational state that they recognize. The next couple of slides show that these types of studies that I'm showing you are also relevant to the study of viruses and other pandemics, including Ebola virus, where we map the regions of the Ebola spike that are recognized by the ZMAP cocktail of antibodies. And on the next slide, for the 2009 influenza pandemic strain, where we map the binding of broadly neutralizing antibodies close to the membrane surface and worked in close collaboration with researchers in New York to work towards developing a strain that might be helpful for universal flu, as a universal flu vaccine. Lastly, in the next slide, I want to mention the importance of understanding mechanisms underlying cell-to-cell transmission. These are obviously early days in the study of SARS-CoV-2 transmission, but in the case of HIV transmission, 3D microscopic imaging studies like what we did then provided many unexpected insights into the intricacies of how viruses are transmitted at sites of cell-cell contact, and also demonstrated how the virus hides in membrane folds of antigen-presenting cells, which, if you advance to the next slide, should show up as an animation. These are tiny viruses that can sometimes be occluded on surfaces of cells, and these kinds of studies will be undoubtedly important in the future. My final slide, I'd like to summarize by noting that cryo-EM imaging methods provide powerful tools for deciphering viral entry and transmission mechanisms, and that we welcome collaborations with academic and industrial partners that could help accelerate the discovery of effective therapeutic routes or intervention. Lastly, I'd like to acknowledge in the next slide a Canada Excellence Research Chair Award from the Canadian government, as well as support from the VGH Foundation, and especially a new award from Genome BC to support our work. Thank you very much.
Thank you, Stuart. We're going to switch topics a little bit, and now we're talking about vaccines. We have Will Jeffries and Leonard Foster talking about one approach, and that's followed by James Taylor from Division Analysis. So, Will?
Okay, good afternoon. I'm Will Jeffries, and my colleague and main UBC collaborator on this project is Leonard Foster, and we're doing a tag team today, so good. Let's see how that goes. So health officials have told us the coronavirus COVID-19 pandemic may continue until an effective vaccine is created that halts transmission, and we've been funded recently by the Michael Smith Health Research Foundation to create candidate COVID-19 vaccines and to create tools for immunological profiling of patients to assess if they're mounting a protective immune response against the virus. So for many years, both Leonard and I have worked on vaccine design, construction, and testing. And I'm even old enough to have worked on the vaccines for the 2004 SARS coronavirus pandemic, and more about that in a little bit. More recently, we created and tested a vaccine for West Nile virus and tested it in the Level 4 facility in Winnipeg. And we've also worked on vaccines for other diseases, including Alzheimer's disease and metastatic cancers. So we've developed these vaccines in multiple formats where the vaccine components are peptides or recombinant proteins or expressed in different vaccine expression systems, including disabled adenoviruses or poxvirus vectors. So over to you, Leonard.
Yeah, and my role in vaccine development has really been to identify candidate antigens for making subunit vaccines, and most of the work we've done so far has been on intracellular bacterial pathogens, but the process that we would use for a virus is essentially the same. Next slide, I think.
So obviously, for the purpose of creating a vaccine, this is a major issue for potential national and international needs to halt the pandemic by halting transmission, and also halting future outbreaks of the disease. Over to you, Leonard.
Yeah, and so in addition to trying to create a vaccine, we also need surveillance tools that would be part of this research, and one of the key ones would be HLA tetramers, which would allow us to evaluate whether a patient is mounting an immune response to the pathogen if they're infected, or in the case of a vaccine, whether they are effectively being protected.
Slide 3, please. Okay, so though there are more than 40 vaccines under development worldwide, and these include many formats using different viral components, but it remains unclear which of the combinations will be successful, so additional candidate vaccines need to be tested until an effective vaccine for COVID-19 is created. So the format, or the primary format of the candidate vaccines we're creating is based on lipid nanoparticle encapsulated mRNA, where we're encoding the viral spike protein, which is a major component of the virus. Subunit vaccines of this type have an advantage in that they have lower health risks, and mRNA LNP formats have an additional advantage in the speed in which they can be constructed and tested. And the fact that the candidate vaccines created in this format can be mass-produced in cell-free conditions using good manufacturing practices that meet the quality control and standards for inclusion in clinical trials. So the vaccines we're concentrating are vaccines where we've engineered adjuvant components within the vaccine which boost the immune response and enhance long-lasting immunity at very low vaccine doses compared to standard vaccines. And we plan to test the candidate vaccines in preclinical models in the National Microbiology Laboratory in Winnipeg, where they've tested other vaccines, including vaccines for Ebola virus, or at UBC in the Kinder facility. We will assess the performance of the candidate vaccines cross-test against different strains of viruses. This may be a very important feature going forward to eradicating the virus and halting the pandemic through halting transmission. And I think I need to stress that these unique candidate vaccines that we're testing have the potential to overcome a major obstacle in vaccine development for large global populations by reducing the dosage of the vaccine that's required for complete protection. And this will increase the speed and reduce the cost for producing and manufacturing vaccines. So again, over to you, Leonard.
Next slide, please. So the role that we'll play is in, as I said, identifying the epitopes. And the way we do that is to take cells that are infected with virus or pathogen of whatever sort, and then identify using mass spectrometry the peptides that are presented on the surface of those cells on the MHC molecules. And then through that process, we can then use those epitopes to create the HLA tetramers that would be used for monitoring immune status of infected or vaccinated individuals. Next slide.
Deliverables we're planning to share with the community. And so this includes the distribution of monitoring tools for personalized immune profile in patients infected with the virus. And we also plan to update the community on our progress in validating the COVID-19 vaccines in preclinical models prior to clinical testing. And we're also seeking now collaborators for testing these tools. So unfortunately, when the 2004 SARS coronavirus was deemed no longer a threat, the funding for coronavirus vaccines quickly disappeared. Thus, we exited the 2004 SARS pandemic having learned very little about the creation of effective vaccines for coronaviruses in general. So we hope that the lessons we learned regarding creating an effective vaccine for COVID-19 will inform vaccine design during the current global pandemic, as well as the next one. Thanks for your attention.
Good. Well, that's actually a very nice lead into James Taylor's presentation of precision nanosystems. We're developing much of the lipid particles.
Fantastic. Well, thank you for the opportunity to speak today. So next slide. So just to background Precision Nanosystems, so we are a UBC spinout, about a decade old, and we're a leader in providing manufacturing technology for gene therapies, advanced therapeutics, and our technology is supporting drug development work by over 120 biopharma companies, over 100 academic institutions as well. And over 15 of our users are now developing COVID-19 vaccines. We are all about nanoparticle and nanomedicine, so that the previous talk was fantastic. Lipid nanoparticle, polymer nanoparticle systems, and they're used to package and deliver drugs to take the drug active ingredient directly to cells and tissues to have their effect. Our two core technologies are a continuous flow manufacturing technology based on our proprietary microfluidic platform, and also nanoparticle delivery technology itself for nucleic acid-based delivery. Vancouver is a real hub for nanomedicines, a center of excellence, and it's fantastic to be a part of the community here. Next slide, please. Our platform really enables solutions across the full drug development paradigm, from the earliest stages of invention and screening and development all through to clinical manufacturing. And we try to provide complete solutions for our users to enable them to make transformative medicines, from the manufacturing with our instrumentation, delivery technologies through our reagents, and clinical
delivery platforms for working with our partners, as well as our expertise to provide any of the expertise that's required to develop these important and sophisticated therapeutics. Next slide, please. Today I want to talk about the power of genetic vaccines. This is RNA or DNA vaccines. And it really is enabling technology that practically only coming around in the last 5 years or so, but allows us to very rapidly develop vaccines across to pathogens. And this could be known or unknown pathogens. And so once we have the sequence of a pathogen, we're able to understand very rapidly with bioinformatics now understand the antigens that could be produced from that sequence, the protein antigens that could be produced. We can put that into an RNA cassette. That RNA cassette then packaged into a nanoparticle construct where that nanoparticle delivers that RNA into the appropriate cells in the body, where then the RNA expresses proteins using the cell as a factory and expressing that antigen. And unlike typical vaccine production, this can be done in the matter of weeks. And we're seeing many of the leading COVID-19 vaccine platforms being RNA-based, based on the speed and the efficacy of these types of vaccines. So next slide, please. Ourselves are collaborating with researchers globally around RNA vaccine constructs as well, using our delivery platform. And this is, we're primarily focused, many great approaches, our approaches in what we call self-amplifying RNA vaccines. And similarly, again, these are synthetic vaccines, so we're able to synthesize the RNA construct. We're able to rapidly package that into a nanoparticle system that can deliver to the appropriate cells in the body, administer that to patients. The RNA expresses the antigens, and you get a potent immune response. The self-amplifying RNA approach allows for very potent dosages. So in the order of a liter of drug product manufacturing, we can get in the millions of human dosages. These systems, as all RNA systems, require a lipid nanoparticle or a nanoparticle delivery system. It requires a scalable nanoparticle manufacturing platform, but once established, these concepts can be applied to today's pandemics or future pandemics, where the raw materials are generic. It's really the information, the sequence that is different, and we can very rapidly respond to any new issue or changes that may happen with genetic drift and the like. Next slide, please. And lastly, just as a manufacturing platform company and a leader in this field and Canadian, we feel it's very important to be bringing in Canada manufacturing platforms and enabling these. And we're leading an effort right now to put together all the requisite technology, our platform and other platforms and companies to help lead the ability to make these, develop these drugs in Canada quickly and to enable future manufacturing of these drugs directly in Canada. And that's important for many, many reasons. But we are looking for partners. We're looking for all partners to help us with this. We think it's important. We're very passionate about it. And our company is extremely passionate about enabling this field and bringing these therapeutics and vaccines into patients. So thank you very much for the opportunity to speak today, and it's been a fantastic conference.
Thank you, James. So we're now hearing from Lindsay. So we've heard about Finder for the last several talks, and Lindsay's going to give a little bit broader scope on what Finder is at the Life Science Institute at UBC.
Thanks, Rob. So my name is Lindsay Elsis. I'm in the Department of Microbiology and Immunology, but I'm here speaking with you today in my capacity as the Scientific Director of Finder. As been already mentioned, Finder is an acronym for the Facility for Infectious Disease and Epidemic Research. If you are planning to do any research involving isolating, propagating the virus, then you'll probably want to get in touch with us, and you'll also get to know the head tech and manager of Finder, who is Dean Airy. Next slide, please. So Finder is a CL3 facility. used for tuberculosis, influenza, and other emerging diseases. As has already been mentioned this afternoon, it was actually founded by Francois Jean. He spearheaded a CFI grant that was awarded back in 2004. So we all owe him a debt for really his vision of putting this together. Finder uses or really enables cutting-edge approaches to not just to understand the biology of RG3 pathogens, but also to develop novel therapeutic strategies and vaccines. It's got a lot of infrastructure. It's essentially an ensemble of custom-designed, self-contained microbiology suites, all of them dedicated to different pathogens, and each with their own animal holding room. And in one of these holding rooms, we've actually got an imaging suite as well that allows for live imaging of small animal models. Next slide, please. So I think the most important considerations about Finder with respect to this community is that it's got a number of unique capabilities in the Lower Mainland. So right now, it's actually the only CL3 facility with a lab that's dedicated to virology research, and it's the only CL3 facility that can support research with animal models. So at this stage, again, I think as Francois alluded to earlier, we don't actually have the virus in Finder yet. So we're at the stage where we're just finalizing revised SOPs, and these are required for PHAC certification. But Francois has actually already got a postdoc in Finder, and the virology lab, which has sort of lain fallow for a few years, is now operational again. 3 other researchers have been funded to work in Finder already, so we're getting going. They all spoke this afternoon, Horatio, and Eric, and Joseph. And so again, I'm just letting you know this facility is here. Will allow you to do some very unique things. If you're interested in using it, please contact Dean Airy. We will assist you in developing SOPs as well as training your people so that you're able to work in the CL3 facility. That's all I have to say. Thanks very much.
Thanks, Lindsay. So thank you for all the speakers in this group. As you can see, the diversity of the innovation that's really being applied. And I'm just reflecting here, 4 months ago, we hadn't heard of COVID-19. And here we've had now, we've not bad, we're only about an hour overtime from our estimated time of having, you know, the expertise that's come to address COVID is, I think, is remarkable, and certainly put BC from all aspects, really as leaders for both COVID management and COVID research. So and David will have some concluding remarks.
I'd have to agree, Rob. I mean, I think I can actually pull these things together in about 2 minutes. When we started talking, we mentioned that the reason we need research like this is because we need to find a way to put an end to the threat of COVID-19 to the people of BC and the world, really. And we had said that that may require either a lot of innovation in public health methods to drive transmission down to zero, potentially a vaccine, or we could be helped along by therapeutics, and even if they can't stop all transmission, they could stop an awful lot of the morbidity. So we started off hearing about how diagnostic testing and genomics were critical to informing both epidemiology and modeling, and how those sciences are informing public health action. We learned how that public health action can be improved with things like mobile health tools, by having a much better understanding of mental health, gender, cultural, trust, and information implications of what we're doing. And these relate nationally and internationally to things like travel. So we had an awful lot of solutions pointing towards what we can do for public health measures. The viral genomics also gets together with the developing immunology towards vaccine development. We heard of the innovative employment of cryo-EM, HLA tetramers, mRNA vaccine design, nanoparticles to that field. And we also heard how Canadian clinical scientists are ready to test vaccines as well. Immunology is also informing development of immunotherapeutics, and we heard from the Bach Lab and Accelera about some areas there. So we're moving ahead with immunology and contributing to vaccine development. On the drug discovery side, I like Francois' approach to saying, yeah, we've got repurposed drugs, we've got drugs that focus on host factors, and we've got drugs that are in essence novel antivirals, and we heard about small molecule screening, proteomics, use of organoids, and again, some of the technologies we've already mentioned. And as we get those drugs online, we've also learned that we have a robust international clinical trial structure with which to evaluate them. So we're really hitting those three key areas that can provide answers that might help end this. We also learned that we have shared infrastructure, both informatic infrastructure and the BSL-3 labs at the LSI, the Finder labs. And I particularly liked to hear the note, which is dear to my heart. We are actually, as a research community, contributing to the response in BC. We actually coordinate with the people planning the response in government. And I liked to hear the advanced thinking around what about the supply chain? Because if we have vaccines or drugs developed that are going to help us this way, we better be able to manufacture them and get them to our people in a timely fashion. So that's how I'd sum it up. You're hitting all three key areas of pandemic response right on the head.
I think, David, that really summarizes the afternoon and our challenge for the next years to come on this. I think we will probably plan another one of these. We also want to have one for public, more of a public outreach, and really looking for ideas how to keep this collaboration and sharing of information moving forward. We put a website down just for this conference. UBC has a new website on collating a lot of the grant information on COVID, and certainly the CDC has a wealth of information on the pandemic side. So I can't thank we still have about 400 people on, everybody for participating, and all the speakers. I think it's been a fantastic afternoon. So thank you to all, and we'll be forming more of these as the time goes on. Thank you. Good. Stay safe, stay apart. Happy Easter. Okay, thanks, everybody.